Hello, it's Dmitry again. Today we'll be covering the first aid for the USMLE Step 1 textbook 2025 edition, the Microbiology chapter. Now, I've read this chapter before in the past. I've never streamed it, and I've read it pretty far back in the past, and so I'd like to review it, of course, leading up to the board exam. And so I'll do that today. I'll be only reading the first part, the first half or so, you could say, of the chapter today. It is a relatively long chapter. I'm on page 121 of my textbook. I'll be going to page 199 for the entire chapter, so I'll probably cut that into two different videos. So, bear in mind, again, my book that I'm reading from in person is a 2025 edition. It is the latest edition. However, my PDF that I'm showing you on the screen right now is a 2017 edition, so the text will be somewhat different, although generally speaking, the content will be very similar. So let's begin with a few different quotes. And I'll be reading my quotes. Within 1 linear centimeter of your lower colon, there lives and works more bacteria, about 100 billion, than all humans who have ever been born. Yet many people continue to assert that it is we who are in charge of the world. Neil deGrasse Tyson. Now, I was originally reading the beginning of the quote right there with that, and I was thinking like, oh, this is going to be a quote that actually relates to the topic at hand. That's not necessarily just completely unrelated, just happens to mention the word microbiology. Well, that was kind of true, and then just kind of threw that sort of this is like something he would post on Twitter. Like, what's a Neil deGrasse Tyson post on Twitter? They post like almost like every year or something. It's like something about looking in a mirror or something like that. It's just sort of goofy kind of understanding of whatever, but okay. It's sort of like that pop science. Oh, well, that really makes you think, right? Wow. Okay, whatever. What lies behind us and what lies ahead of us are tiny matters compared to what lies within us. Henry S. Haskins. Wise and humane management of the patient is the best safeguard against infection. Florence Nightingale. Okay. I sing and play the guitar, and I am a walking, talking bacterial infection. Kurt Cobain. Okay, whatever. Anyway, more importantly, let's read this below. Microbiology questions on the Step 1 exam often require 2 or more steps. Given a certain clinical presentation, you will first need to identify the most likely causative organism, and you will then need to provide an answer regarding some features of that organism or relevant antimicrobial agents. For example, a description of a child with a fever and a petechial rash will be followed by a question that reads, from what site does the responsible organism usually enter the blood? This section, therefore, presents organisms in 2 major ways, in individual microbial profiles and in the context of the systems they infect and the clinical presentations they produce. You should become familiar with both formats. When reviewing the systems approach, remind yourself of the features of each microbe by returning to the individual profiles. Also be sure to memorize the laboratory characteristics that allow you to identify microbes. Yeah, so that's the issue. It's not just that you need to know, for example, like, oh, well, Neisseria is a diplococci, and Staph aureus is, catalase positive, and you need to know all the little factoids about each individual organism that allows you to distinguish it from everything else. But you also need to know what they do, what they cause, what's a clinical presentation, what are some of the consequences of them of an infection, that kind of thing. So that because they won't ask you necessarily super directly, they might ask you like, okay, the organism, has this clinical presentation, does X, does Y, has encephalitis, they have meningitis, they have this, and then they talk about maybe they'll say, okay, and it's a Diplococcus species or something like that. They'll give you some identifying information that helps you figure out, oh, this is what it is. And then they'll ask you something like, and what's another complication that might pop up? And they have to say, oh, it's, something like hemolytic anemia, that's a possibility. It's going to be some sort of extra thing where you have to kind of like know everything about it without necessarily them telling you the name. And the answer is not going to be the name of the species. It's going to be some other thing about the same organism. So you have to have a pretty good understanding of each individual organism or how they exist within the context of a broader family of organisms like, Staph species, Streptococcus species, etc. Anyway, let us continue. Microbiology, basic bacteriology. Microbiology, basic bacteriology, bacterial structures. So the structure, the chemical composition, and the function. Appendages, flagellum, which are composed of proteins, and they're used for motility. The pilus, and fimbriae are glycoproteins, or made of glycoproteins, and they're used to mediate adherence of bacteria to the cell surface. Sex pilus are used during conjugation. True. Specialized structures. Spore. Keratin-like coat. Dipicolinic acid. Peptidoglycan. DNA. Survival. Resist dehydration. Heat. Chemicals. Cell envelope. So capsule. Capsule is composed of discrete, usually made of polysaccharides, and rarely is made up of a discrete layer, usually made up of polysaccharides, and rarely proteins. Capsules are used to protect against phagocytosis. The slime layer, loose network of polysaccharides, and they are used to mediate adherence to cell surfaces and play a role in biofilm formation like indwelling catheters. Outer membrane. Outer membranes are made up of an outer leaflet containing endotoxin, LPS, or LOS, which is lipopolysaccharide or lipooligosaccharide. Embedded, and it's like for gram-negative stuff typically, right? If you have LPS, it's gram-negative specific. LOS you might find in something like Neisseria gonorrhea. Embedded proteins, porins, and other outer membrane proteins, OMPs, and then an inner leaflet which contains phospholipids. Now these are gram-negative only, and so that goes back to what I was saying before. They're used as an endotoxin. Lipid A induces TNF and IL-1, antigenic O polysaccharide component. Most OMPs, outer membrane proteins, are antigenic. Porins transport across outer membranes. What about periplasm? Periplasm is a space between the cytoplasmic membrane and the outer membrane and gram-negative bacteria. Peptidoglycan in middle. They accumulate the components exiting the gram-negative cells, including the hydrolytic enzymes like beta-lactamase. What about the cell wall? Cell wall is a peptidoglycan Cell wall is made of a peptidoglycan, which is a sugar backbone with peptide side chains cross-linked by transpeptidase. It's used as a net-like structure, giving rise to a rigid support. Protects against osmotic pressure damage. And finally, a cytoplasmic membrane is a phospholipid bilayer sac with embedded proteins, penicillin-binding proteins, PBPs, and other enzymes. Lipotocoic acid, gram-positive, only extends from membrane to exterior. And it's used as a site of oxidative and transport enzymes, PBPs involved in cell wall synthesis. Lipotocoic acids induce TNF-alpha and IL-1. Cell walls, and I have my thing's the cell envelope, but it's showing you the difference between gram-positive and gram-negative. You can see that layer for gram-positive. Let's talk about that first. So they both have a cytoplasmic membrane. They both have a peptidoglycan, but the peptidoglycan is a lot thicker and multi-layered in the context of gram-positives. There are these things called flagellum and pili, and that kind of thing that both have, and a capsule, which both have. However, there's some things called lipotocoic acid, which you will find in the gram-positives, while there's these things called there's an LPS endotoxin layer, also called, in some cases, an LOS for like Neisseria species. And that's going to be that additional layer that exists for gram-negatives. So gram-negatives have a secondary layer, and they have their peptidoglycan layer is a lot more limited compared to the gram-positives, which are more complicated in terms of peptidoglycan layer. And they're both gonna have flagellum, they're both gonna have pili. You're gonna have these porins on that LPS layer as well for gram-negatives. What about staining? Bacterial taxonomy. So I'm gonna go to stains actually. I'm gonna see if I can find that here. Here we go. So stains, gram stain. First line lab tests and bacterial identification. Bacteria with thick peptidoglycan layer retain crystal violet dye, gram-positive. Bacteria with thin peptidoglycan layer turn red or pink, gram-negative, with counter stain. These bugs do not gram stain well, these little this is a mnemonic here. These bugs do not gram stain well. These little microbes may unfortunately lack real color but are everywhere. So this is mnemonic. So you said these little microbes may unfortunately lack real color but are everywhere. So that's TLMMULRCBRE. So Treponema, Leptospira, these are too thin to be visualized. Mycobacteria, which is cell, has a high lipid content, which prevents it from being properly visualized with staining. Mycoplasma, ureaplasma, which they have no cell wall. Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, Ehrlichia. These are primarily intracellular. Chlamydia lacks classic peptidoglycan because of the decrease in muramic acid. What about GM cystine? GM cystine, H. pylori, Chlamydia, Borrelia, Rickettsia, Trypanosomes, Plasmodium. Another thing here says as a sort of learning device, help certain bugs really try my patience. Okay, if you want to use these, you can. Knock yourself out. Whatever. Periodic acid shift stain. Stains glycogen mucopolysaccharides used to diagnose Whipple disease, Tropharema whipplei. Zeal-Nielsen stain, carbofuscin. Acid-fast stain for Mycobacteria, Nocardia. Stains mycolic acids in cell wall, protozoa like Cryptosporidium oocysts. Along with that, auramine rhodamine stain is often used for screening, inexpensive but more sensitive. What about India ink stain? Cryptococcus neoformans and mucicarmine can also be used to stain thick polysaccharide capsule red. What about silver stain? Silver stain, I have a little learning device here as well, but it says here Helicobacter pylori, Legionella, Bartonella, Henselae, and fungi. And also coccidioides, that is to say, for fungi they say coccidioides, Pneumocystis jirovecii, and Aspergillus fumigatus. And they have a little learning device here that says helicopters are silver. Okay. Because Helicobacter, Legionella, Coccidioides, Pneumocystis, and Aspergillus. Okay. Fluorescent antibody stain. Used to identify many bacteria, viruses, Pneumocystis jirovecii, Giardia, and Cryptosporidium. Example is FTBA-BS for syphilis. Okay. And so you can see some pictures right there just that are relevant within the respective locations. You can see for trypanosomes is picture A. B is showing the Tropharema whipplei. C is showing the mycobacteria. D is showing the Cryptococcus neoformans. E is showing the fungi coccidioides. So we're talking about special culture requirements. So I'm not sure if it's going to be further down the field or not. Yeah, right here. Special culture requirements. Bug, media used for isolation, and medial content / other. So H. influenza, you use a chocolate agar for H. influenza. Or, Haemophilus influenza. Media contents and other / other, factor 5, NAD+, and factor 10, hemotin. For chocolate agar. The Neisseria, gonorrhea, and meningitis. These use a Tyre-Martin agar. Selectively favors growth of Neisseria by inhibiting the growth of gram-positive organisms with vancomycin, gram-negative organisms except Neisseria with trimethoprim and colistin, and fungi with nystatin. Very typically cultured Neisseria. Okay, whatever. Just remember that Tyre-Martin is for Neisseria species. B. pertussis. You would use Bordet Gengu. I would move. Is that Gengu or Gengu? Or Genju? Or Genju? Bordet Gengu. Agar. Bordet for Bordetella. Reagan low medium. Okay. Potato extract, charcoal, blood, and antibiotic. C. Diphtheria. Corynebacterium diphtheria. Use a Tellarite agar and a Loeffler medium. Mycobacterium tuberculosis. Lowenstein Jensen medium, Middlebrook medium, rapid automated broth cultures. Mycoplasma pneumonia, use an Eaton agar and requires cholesterol. For lactose fermenting enterics, use McConkey agar. Fermentation produces acid, causing colonies to turn pink. E. coli, eosin methylene blue, EMB agar. Colonies have green metallic sheen. Brucella, Francisella, Legionella, Pastorella. Buffered charcoal yeast extract agar with cysteine and iron. The Ella siblings, this is another learning device here. The Ella siblings, Bruce, Francis, a Legionnaire, and a Pastor, okay, built the cysteine chapel out of charcoal and iron, okay. Okay, sure. Well, again, some of these things seem a little bit over the top for me because I feel like at some point you just want to remember what these things are rather than remembering a billion different kinds of learning devices. But whatever. Brucella, Francisella, Legionella, Pastorella use charcoal, cysteine, iron. Remember that Legionella and Pastorella are definitely using those because Brucella doesn't matter as much as far as I'm aware. Francisella not as much. Legionella and Pastorella, definitely no Legionella charcoal yeast extract thing. What about anaerobes? For anaerobes, examples include Clostridioides, Clostridium, Bacteroides, Fusobacterium, and Actinomyces israelii. They lack catalase and or superoxide dismutase and are thus susceptible to oxidative damage. Generally foul-smelling, short-chain fatty acids are difficult to culture and produce gas in tissue, carbon dioxide, and dihydrogen. This is another learning device. Anaerobes can't breathe fresh air. So that's CBFA for the... Let me go down a little bit more. For the Clostridium, Bacteroides, Fusobacterium, and Actinomyces. Anaerobes are normal microbiota in the GI tract, typically pathogenic elsewhere, causes aspiration pneumonia. Aminoglycosides are ineffective against anaerobes because these antibiotics require O2 to enter into the bacterial cell. Facultative anaerobes may use oxygen as a terminal electron acceptor to generate ATP, but can also use fermentation and other O2 independent pathways. Streptococci, Staphylococci, and enteric gram-negative bacteria. What about intracellular bacteria? Intracellular bacteria. Obligate intracellular. Rexedia, Chlamydia, Coxiella rely on the host ATP. They stay inside cells when it is really chilly and cold. Facultative intracellular. Salmonella, Neisseria, Brucella, Mycobacterium, Listeria, Francisella, Legionella, Yersinia pestis. Another learning device here would be some nasty bugs may live facultatively. Okay. Encapsulated bacteria. Examples are Pseudomonas aeruginosa, Streptococcus pneumonia, Haemophilus influenza type B, Listeria meningitidis, E. coli, Salmonella, Klebsiella pneumonia, and Group B strep. Their capsules serve as an antiphagocytic virulence factor. Capsular polysaccharide plus or minus the protein conjugate can serve as an antigen in vaccines. A couple polysaccharide antigens alone cannot be presented to T cells. Immunogenicity can be enhanced by conjugating capsule antigens to a carrier protein. Again, here's a little learning device. This is please shine my skis. Are opsonized and then cleared by spleen. Asplenics, that is to say no spleen here, have decreased opsonizing ability and thus increased risk for severe infections. And need vaccines to protect against Neisseria meningitidis, strep pneumonia, and H. influenza. Okay, what about urease positive organisms? Urease positive organisms. Proteus, like Proteus mirabilis, Cryptococcus, Helicobacter pylori, Ureaplasma, Nocardia, Klebsiella, S. epidermidis, Strep epidermidis, Staph epidermidis, and Staph saprophyticus. Urease hydrolyzes urea to split into ammonia and carbon dioxide, leading to an elevation in pH, predisposes to struvite, magnesium ammonium phosphate stones, particularly Proteus mirabilis will do that. And another little mnemonic here, learning device, P chunks. Sure. Okay. Yeah, whatever. Next one is catalase positive organisms. Catalase positive organisms. Catalase degrades. What is that technically called? I want to make sure I'm not saying anything incorrectly here. Let me see. Okay. Sorry, hydrogen peroxide. So anyway, catalase degrades hydrogen peroxide into water and bubbles of O2. You can see in the picture. Before it can be converted to microbicidal products by the enzymes myeloperoxidase. People with chronic granulomatous disease and NADPH oxidase deficiency have recurrent infections with certain catalase-positive organisms. Catalase-positive organisms include Candida, Pseudomonas, Nocardia, Bordetella pertussis, Burkholderia ipsipacia, Helicobacter pylori, Aspergillus, Staphylococci, Serratia, Listeria, E. coli, catalase-positive, notoriously big bubbling hassle. Okay. Sure. So that's the little learning device I have at the end. Your thing says cats need places to belch their hairballs. Well, that one's definitely worse. So they definitely improved their learning device since 2017. But take it or leave it, in my opinion. I don't find them either to be particularly memorable. My biggest recommendation is, this is actually an important one, is just simply remember the list of things you have to have on that list. Candida, Pseudomonas, Nocardia, Bordetella, Burkholderia, Helicobacter pylori, Aspergillus, Staphylococcus, Serratia, Listeria, E. coli. A lot of big names. So remember that. Then if you have an infection with those kinds of things, I keep on having these infections of these organisms that are catalase positive, maybe you have an NADPH oxidase deficiency. Maybe you have CGD, right? If you have a question stem like that, a silhouette talking about a patient who's 7 years old and they have all these sorts of infections, and they start listing them out, and they're like these kinds of things. You say, oh, well, maybe it's CGD. And you have to explain what does CGD even mean. It's like, oh, well, they have an NADPH oxidase deficiency. You say, oh, wow, cool. Just know your stuff. Just know all the information. Just know all information. Just know all medical information. You'll be fine for the board exam. Pigment-producing bacteria. Let me see. Pigment-producing bacteria. Actinomyces israelii yellow sulfur granules, which are composed of filaments of bacteria. Israel has yellow sand. Okay. Staph aureus, yellow pigment. Aureus means gold. Yes, aureus means gold in Latin. P. aeruginosa, blue-green pigment, pyocyanin and pyoverdin. Arugula is green. Okay, sure. And remember that pyoverdin is related to the iron. I forgot what pyocyanin is related to, but it does some other thing. But yeah, remember that the Pseudomonas has those two pigments, which cause it to have that kind of greenish color. Serratia maricens, red pigment. And it's going to say, think red sriracha sauce. Okay, sure. In vivo biofilm producing bacteria. Here's a little tip and trick for you. I'm sure the people who wrote this textbook thought some of these ideas that were for learning devices are really clever and unique and helpful. For a lot of people who are reading it, probably that's not necessarily true. My recommendation is that if you have difficulty remembering something, make your own learning device. Come up with your own little learning device. You think, I need to know all the species that cause issues with CGD. Just come up with your own learning device. Just make up something. Think about what are the first letters, and then maybe make an acronym. Maybe make some sort of other little gimmick or whatever. Think about it, right? If it's important, if it's high yield, maybe consider, and it's a lot of sort of rote memorization, just think about how you can remember that on your own terms. This makes it easy for you to remember personally, because what you find is an important reference point personally might be different from what a person would find to be memorable for somebody else. So bear that in mind. In vivo biofilm producing bacteria. Staph epidermidis, and then catheter and prosthetic device infections. Very true, very true. Viridon streptococci, strep mutans, and strep sanguinis. Dental plaques, infective endocarditis. True. Pseudomonas aeruginosa, respiratory tree colonization in patients with cystic fibrosis, ventilator-associated pneumonia, contact lens-associated keratitis. What about non-typable unencapsulated Haemophilus influenza, otitis media, middle ear infection? Okay, now we're going to go into spore-forming bacteria. Oh, what was that? Okay, here we go. So I can flip between pages. Where was I? Spore-forming bacteria. I'm getting the impression that the older version of this textbook is a lot more detailed than this one. And this one's already very, very long, so... Okay, so spore-forming bacteria. Some gram-positive bacteria can form spores when nutrients are limited. Spores lack metabolic activity and are highly resistant to heat and chemicals. Core contains diplocalinic acid, responsible for heat resistance. Must autoclave to kill spores, as it is done to surgical equipment by steaming at 121 degrees Celsius, which is pretty hot. Oh, okay, so that's beyond boiling, for 15 minutes at a pressure of 15 psi. Very important. Hydrogen peroxide and iodine-based agents are also sporicidal. So bear that in mind. It's not that it has to be simply hot or for 15 minutes. It has to be high pressure, high heat, long-term environment, and that'll kill the spores, because spores are very, very hardy. Examples for spore-forming bacteria: Bacillus anthrax, Bacillus cereus, food poisoning, causes rice water. Oh, no, this causes rice water. It causes diarrhea, but it's from reheated rice. Clostridium botulinum, botulism. Clostridioides difficile, pseudomembranous colitis. Clostridium perfringens, gas gangrene. Clostridium tetani, tetanus. Autoclave to kill Bacillus and Clostridium, ABC, sure. That is true, though. Clostridium species and Bacillus species are very hardy, so they form spores. They are spore-forming species, or genuses, genera. Anyway, bacterial virulence factors. Are we sure we didn't miss this before? Okay. Bacterial virulence factors. These promote evasion of host immune response. Capsular polysaccharide, highly charged hydrophilic structure, acts as a barrier to phagocytosis and complement-mediated lysis. Major determinant of virulence. Protein A. Protein A binds to the Fc region of IgG, prevents opsonization and phagocytosis, expressed by Staph aureus. IgA protease. Enzyme that cleaves IgA, allowing bacteria to adhere to and colonize mucous membranes, secreted by strep pneumonia, Haemophilus influenza type B, and Neisseria. What about M protein? Helps prevent phagocytosis, expressed by group A strep, which is to say strep pyogenes. Sequence homology with human cardiac myosin, molecular mimicry, possibly underlies the autoimmune response seen in acute rheumatic fever. I mean, possibly, very likely, yes. Now let's talk about the antibiotic resistance mechanism. So we'll skip this. Go down here. Do we have that? I don't know. Do we? We might not have it, so I'm just going to describe it for you a little bit. Antibiotic resistance mechanisms. I think this diagram is probably pretty good. There's enzymatic inactivation, decreased efflux, decreased uptake, and the third category is drug target modification. So let's talk about the first category. We have enzymatic inactivation. So these are about basically, these are mechanisms that the bacteria can use to prevent itself from being killed by antibiotics or being inhibited by them some manner or way. So enzymatic inactivation, drug cleavage, penicillin G and V, penicillinase-sensitive penicillins, cephalosporins, carbapenem. So these are basically antibiotics that are susceptible to these kinds of antibiotic resistance mechanisms. Drug modification. So we're talking about enzymatic inactivation. Drug modification, aminoglycosides, chloramphenicol. What about increased efflux, decreased uptake? Tetracyclines, sulfonamides, and fluoroquinolones are susceptible to these. And what about drug target modification? So for these ones, we're talking about rRNA, so like linezolid, macrolides, enzymes like penicillin-resistant penicillins, cephalosporins, sulfonamides, and fluoroquinolones. And then finally, cell wall. Cell wall can help prevent the entry of vancomycin, which is pretty basic, right? Now we're going to be talking about bacterial genetics. I'm going to go take a short break. Okay, I'm back. And I think we're gonna probably wrap up with the parasitology section today, and do virology in the next session. So we'll wrap up on page 159, and we're currently on page 129, I believe. Sorry, 128. So let me go back to the page that's relevant. Here we go. Bacterial genetics. So these are different kinds of methods of bacterial genetics in terms of like the gene transfer and that kind of thing. So bacterial genetics, horizontal gene transfer is the main mechanism for transfer of antibiotic resistance among bacteria. So transformation. And you can see the little chart right there. Competent bacteria can bind and import short pieces of environmental naked bacterial chromosomal DNA from bacterial cell lysis. The transfer and expression of newly transferred genes is called transformation, a feature of many bacteria, especially Strep pneumonia, Haemophilus influenza, type B, and Neisseria. Shin, that's the little thing they do again. Okay, why not? Adding deoxyribonuclease degrades naked DNA, preventing transformation. So basically you can prevent it from happening by just destroying the DNA before they can take it up into themselves. What about some other methods? Let's talk about conjugation for a little bit. Conjugation, F positive times F negative. Well, it's not really times, it's just the interaction betwixt the two. F positive, fertility factor, plasmid contains genes required for sex pilus and conjugation. Bacteria without this plasmid are termed F negative. Sex pilus on F positive bacterium contacts the F negative bacterium. A single strand of plasmid DNA is transferred across the conjugal bridge, mating bridge. No transfer of chromosomal DNA. Now, they use that terminology, and I'll go into a little bit more later on. They use a terminology, they say sex pilus, and they say mating bridge, for example. It's not sexual reproduction. These are asexual organisms. They do not reproduce sexually. They do not have, as you can imagine, they do not have genital organs or something like that. They do not have intercourse. There's nothing like that per se. But they do exchange DNA with each other, basically. So that's the difference between the two. So basically, there's no sexual reproduction going on, but they're analogizing basically gene transfer to such a thing because it is a method in an asexual species to basically create new genetic diversity. Not to mention the fact that it is also, again, the injection of DNA into a new organism, which, in fairness, you can to be fair, you can analogize all sorts of things to the act. Anything that involves a receptacle and a vaguely cylindrical object could be analogized to that, obviously. But is it actually sex? No, it's not. It's its own thing, basically. They like comparing it, but it is different. It is distinct. HFR and F negative. So F positive plasmid can become incorporated into the bacterial chromosomal DNA, termed high frequency recombination (HFR) cell. Transfer a leading part of the plasmid and a few flanking chromosomal genes. High frequency recombination, it may integrate some of those bacterial genes. Recipient cell remains F negative, but now may have new bacterial genes. So here's the F positive cell with the F negative cell. This is plasmid incorporates into bacterial DNA, and there's a transfer and replication part of the chromosome. And then you have this HFR cell, and you have the recombinant F cell. Okay. Again, those are similar, but not quite the same. But both are forms of conjugation. What about transduction? Transduction, generalized, a packaging error, quote unquote. Lytic phage infects a bacterium, leading to a cleavage of the bacterial DNA. Parts of bacterial chromosomal DNA may become packaged in phage capsid. Phage infects another bacterium, transferring these genes. So we have a lytic phage, bacteria, cleavage of the bacterial DNA, bacterial DNA package in phage capsid, leading to release of new phage from the lyse cell, leading to it infects the other bacteria, and the gene transferred to the new bacteria. Okay. Sure. Specialized, an excision event. Lysogenic phage infects bacterium. Viral DNA incorporates into the bacterial chromosome. When phage DNA is excised, flanking bacterial genes may be excised with it. DNA is packaged into the phage capsid and it can infect another bacterium. Genes for the following 5 bacterial toxins are encoded in a lysogenic phage. ABCDs as a learning device. Group A strep, which again is strep pyogenes. Erythrogenic toxin, botulinum toxin, cholera toxin, diphtheria toxin, Shiga toxin. So you can see the lysogenic phage, the viral DNA in bacteria. Viral DNA incorporates into the bacterial DNA. Phage particles can carry bacterial DNA. Release of new phage from lyse cell, and then infects another bacterium and genes different from the donor and recipient. Okay. So what about bacterial genetics? Bacterial genetics continued transposition. Transposition is a jumping process involving a transposon, specialized segment of DNA, which can copy and excise itself and then insert into the same DNA molecule or an unregulated DNA, for example, a plasmid or chromosome. Critical in creating plasmids with multiple drug resistance and transfer across special species line, for example, TN1546 with Van A from Enterococcus to Staph aureus. Very weird, because when you think about it with non-bacteria, let's say like talking about mammals or something like that, these processes of reproduction are so alien to us because they're all asexual. They're transferring DNA to each other. They're getting, and in all fairness to human beings, they do have viruses that can mess with the DNA, genomes and stuff like that of mammals and of other multicellular organisms. But we're talking about, meaningful changes in phenotype. We're talking about, transfer of genes across species. It's really crazy stuff that's going on in the bacterial world. Compared to what's going on in terms of what's going on like multicellular organisms, which I would based on my understanding is a lot more tightly regulated in terms of genetic information. Because with bacteria, they don't live very long. Viruses don't live these things do not live very long theoretically, or they don't have to, and they reproduce rapidly. And so you can kind of have a lot more leeway with just experimenting and letting errors happen, all sorts of crazy stuff to happen, because the sort of resource investment per organism is not very high. And the generations are very, very quick. So you have a little bit more room for experimentation and new combinations of genes and genomes and DNA transfer and all the rest. Main features of exotoxins and endotoxins. So exotoxins and then endotoxins. So let's talk about exotoxins. They come from certain species of gram-positive and gram-negative bacteria. They are secreted from the cell. They are polypeptides. They are located in the plasmid or bacteriophage. They are highly toxic, fatal dose on the order of 1 microgram. Clinical effects are various, see following pages. Mode of action is various, see following pages. The antigenicity of exotoxins induces a high titer antibodies called antitoxins. Vaccines include toxoids used as vaccines. Okay. Heat stability, they are destroyed rapidly at 60 degrees Celsius, except staphylococcal enterotoxin, E. coli heat stable toxin, and Bacillus cereus emetic toxin. So most exotoxins, with some very important exceptions, are destroyed at 60 degrees Celsius. Typical diseases related to exotoxins: tetanus, botulism, diphtheria, cholera. What about endotoxins? Well, source would be outer cell membrane of most gram-negative bacteria. They are not secreted from the cell. They have a lipid A component of LPS, structural part of the bacteria, released when they are lysed. They are located the genes are located on the bacterial chromosome. Compare that again to the exotoxin. Exotoxins are from the plasmid or bacteriophage. Now the endotoxins are from the bacterial chromosome itself. The toxicity is low. Fatal dose on the order of hundreds of micrograms. Yeah, it's not particularly toxic comparatively speaking. But they can cause fever, shock, hypotension, DIC, which is not good. That could be dangerous. They induce TNF, IL-1, IL-6. They're poorly antigenic. No toxoids are formed and no vaccine is available. They are stable at 100 degrees Celsius for 1 hour. They cause meningococcemia, sepsis by gram-negative rods. Anyway, and there's a little diagram in the bottom that your book does not have, showing exotoxins being released, going downstream by cellular reaction, and these little things called endotoxins, which are part of that overall, remember that layer of the bacteria, of gram-negative bacteria? And they're being broken off, I guess, during lysis, and they're going on to that TNF, they're causing the release of TNF, IL-1, and IL-6. So like exotoxins are like a real deal toxin. And endotoxins are just components of the cell wall, sorry, not the cell wall, but of the layer of gram-negative bacteria, which still trigger reactions, don't get me wrong, but they're definitely a lot less toxic. The endotoxins that we're talking about, that they're more problematic in the sense that they tell the body something's going wrong. When your body knows there's LPS and LOS along, it has this sort of reaction to it that's very, as and it's a sort of immune response to it because it realizes, wait a minute, we're under attack. We have an ongoing infection. So that's more what's going on with that. Exotoxins are a lot more toxic, and so it's more than just the body's response to what's going on when it comes to exotoxins sometimes. If I'm getting that correctly. Yeah, I mean they have various kinds of effects. So just be aware of endotoxins. Part of the wall, or sorry, part of the one of the layers of gram-negative bacteria. It's toxic, but not too crazy. Exotoxins are a little bit more specific, and they are going to cause all different kinds of issues potentially. They can be very deadly, highly toxic, and they're just something that the bacteria will release in some cases. So let's talk about this. Bugs with exotoxins. My book says bacteria with exotoxins. Okay. With the bacteria, toxin, mechanism, manifestation. So Corynebacterium diphtheria has diphtheria toxin. It inactivates the elongation factor 2, EF2, through ADP ribosylation. This leads to pharyngitis with gray pseudomembrane in the throat, and severe lymphadenopathy, bull's neck, and also myocarditis. So none of that's fun. We also have Pseudomonas aeruginosa. The Pseudomonas aeruginosa is called exotoxin A, and likewise it uses the same mechanism. It inactivates the elongation factor EF2 through ADP ribosylation, and that causes host cell death. And there's a little A thing that's a lot of these things has. It says diphtheria toxin, exotoxin A, Shiga toxin. Let me read a little A for you on the bottom right here. It says here, let me scroll down. It says here, an AB toxin, also called 2-component toxin or 3 for anthrax, with B enabling binding and triggering uptake endocytosis of the active A component. The A components are usually ADP ribosyl transferases. Others have enzymatic activities as listed in the chart. So just be aware of that. Now let's talk about Shiga toxin. Shiga toxin, so Shigella species has Shiga toxin. And we also have enterohemorrhagic E. coli. So true. Your book says Shiga-like toxin. I mean, that is fair. They call it Shiga-like toxin all the time. My book doesn't separate the two, but they say, yeah, Shiga toxin, Shiga-like toxin. This toxin that E. coli can have, and Shigella does have, they inactivate the 60S ribosome by removing the adenine from the rRNA. This leads to damage of the GI mucosa, leading to dysentery, toxin-mediated injury, and cytokine release, leading to hemolytic uremic syndrome, HUS, prototypically in the EHEC serotype O157H7. That's for E. coli, by the way. The EHEC E. coli with O157H7 that has Shiga-like toxin. Unlike Shigella, EHEC does not invade host cells. So Shigella will invade the host cells, but EHEC will not. But they both cause HUS. Okay. What about increased fluid secretion? So this messes with protein synthesis. What about increasing the fluid secretion? Enterotoxin. There's a lot of E. coli toxins, by the way. Well, there's a lot of toxins. Some species have more than one toxin. Enterotoxigenic E. coli, heat labile toxin (LT). There's also heat stable toxin (ST). Let's talk about it. Heat labile toxin. It overactivates the adenylate cyclase, increasing cyclic AMP, okay? Increasing the chloride secretion in gut and water efflux. Causes watery diarrhea, labile in the air, adenylate cyclase, stable on the ground, guanylate cyclase. Bacteria that increase cyclic AMP include V. cholera, Bacillus anthracis, B. pertussis, okay. Bordetella pertussis, and E. coli. Heat stable toxin overactivates guanylate cyclase, cyclic GMP increase, leading to a decrease in resorption of sodium chloride and water in the gut. So this is kind of the other toxins. So there's two different kinds of ETEC toxins. There's ETEC toxin that's labile, which is going to be associated with cyclic AMP increasing. There's going to be one that's heat stable, which is going to cause cyclic GMP increasing. Now, They are similar in certain ways, but the way I would remember it would be this. I remember it from the acronym LAGS, 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 which because the labile toxin, think L for labile, increases cyclic AMP, while the heat stable toxin activates cyclic GMP. So LAGS, LAGS. Now, people have their own learning device. You figure it out, okay? Beyond that, it is correct to say that there's other toxins, again, from pertussis toxin, anthrax toxin. Specifically, there's like an edema factor anthrax toxin. There's a lethal factor and there's an edema factor. The edema factor one is one that's related to this, I believe. There's also a cholera toxin, and these all increase cyclic AMP. Now, that's true, but the way they do it is different, so be aware of that as well. One of them will increase G sub S, we'll do it through G sub S. One of them will do it by inactivating through G sub I. So there's like a double negative for the one that does with G sub I. There's one that directly activates adenylate cyclase, and there's another one that actually pretends to be like adenylate cyclase to increase cyclic AMP. So the way they're doing it is different, but they have the same outcome. So just be aware of that. Heat labile toxin, E. coli, that's ETEC. The pertussis toxin, anthrax toxin, edema factor anthrax toxin, cholera toxin, they all will increase cyclic AMP in their own little unique way, of course. What about Bacillus anthracis? Bacillus anthracis has a thing called anthrax toxin, which your book says edema toxin. This mimics the adenylate cyclase, which will increase cyclic AMP, likely responsible for the characteristic edematous borders of the black eschar and the cutaneous anthrax. Now, I'm glad your book calls it edema toxin, even though my book does not call it that, because there is a different thing called lethal toxin, LT, that just kills cells. Okay, this toxin is increasing cyclic AMP. It creates that border. You see, when you see anthrax like lesions, this is the toxin that causes the sort of edematous border. The other toxin, the lethal factor, that'll just kill things. So be aware there's a little bit of a distinction between those two. What about Vibrio cholera? Cholera, again, is one of these toxins that increases cyclic AMP. It says here, overactivates adenylate cyclase, increasing cyclic AMP by permanently activating the G sub S, see what I'm talking about? Increasing the chloride secretion in the gut and water efflux. So true. And this causes us to have a voluminous rice water diarrhea. So this brings us to the next one. It says, inhibits phagocytic activity. Now, pertussis is the double negative one right here. This inhibits the phagocytic ability. So Bordetella pertussis has pertussis toxin. This activates the adenylate cyclase, increasing cyclic AMP by inactivating inhibitory subunit. So basically, it's a double negative. G sub I is inhibiting adenylate cyclase. So if you inhibit the inhibitor G sub I, well then now you're activating the original thing, which is adenylate cyclase, allowing you to have more cyclic AMP. The pertussis toxin causes whooping cough. Child coughs on expiration and whoops on inspiration can cause 100-day cough in adults associated with post-tussive emesis. What about Clostridium tetani? Now we're talking about inhibits the release of neurotransmitters. So there's two different kinds right here, tetany and botulinum. So Clostridium tetani, which causes tetanic, or you might say spastic paralysis. Tetanospasmin is the toxin. These are proteases that cleave snare, soluble NSF attachment protein receptor, a set of proteins required for neurotransmitter release via vesicular fusion. Okay. Well, let's go a little more specific than that. So Clostridium tetani toxin, tetanospasmin, is going to have a toxin that prevents release of inhibitory GABA and glycine neurotransmitters from the Renshaw cells in the spinal cord, leading to spastic paralysis, i.e. tetanic paralysis, rhisis, sardonicus, trismus, lockjaw, and opisthetonos. Opisthetonos. Okay. Whatever, man. And the Clostridium botulinum toxin, that causes, again, it says here, both are proteases that cleave snare, soluble NSF attachment protein receptor, a set of proteins required for neurotransmitter release and via vesicular fusion. This causes infant botulism, caused by ingestion of spores from soil or raw honey, or from canned food, by the way, toxin produced in vivo, foodborne botulism caused by ingestion of preformed toxins from canned food. Okay, from canned food. Okay, so there's infant botulism, which is usually from honey, and there's adult botulism, which is usually from canned food. Now, I want to make this very clear to you. The mechanism, they say, the mechanisms are both proteases that cleave snare. Okay, sure, I don't deny that, but here's the thing. And your book goes into it. One of these causes a tetanic or spastic paralysis because it's preventing the inhibitory molecules GABA and glycine from doing their thing and moderating the response. So you have a spastic paralysis because it can't moderate itself. When you have a botulinum toxin, you have a flaccid paralysis, as your book says, because it's inhibiting the acetylcholine from stimulating things in the first place. So basically, tetanus is inhibiting the inhibitory molecules and the moderating molecules, GABA and glycine, while botulinum toxin is preventing the release of acetylcholine, which is a stimulatory molecule. Hence why it causes against flaccid paralysis. Your book is a little bit better description of that, so I'll talk about it right here. Flaccid paralysis, floppy baby syndrome. Toxic prevents release of stimulatory acetylcholine signals at neuromuscular junctions leading to flaccid paralysis. So just be aware of that. They both cause paralysis, but the means by which they do it is a little bit different. Bacteria with exotoxins, continued. So bacteria, toxin, mechanism, manifestation. So let's talk about lice cell membranes, Clostridium perfringens, alpha toxin. Alpha toxin, phospholipase lecithinase that degrades tissue and cell membranes. Manifestation, degradation of phospholipids, myonecrosis, gas gangrene, and hemolysis, double zone of hemolysis on blood agar. What about Streptococcus pyogenes? Streptolysin O is a toxin. This protein that degrades cell membrane and lyses RBCs contributes to beta hemolysis. Host antibodies against toxin, ASO, used to diagnose rheumatic fever. Do not confuse with immune complexes of post-streptococcal glomerular nephritis. Superantigens causing shock. Staph aureus, Staphylococcus aureus. The toxin is a toxic shock syndrome toxin. Okay, fair enough. What toxin causes toxic shock syndrome? Well, probably the toxic shock syndrome toxin, TSST1. This thing cross-links the beta region of the TCR to the MHC class II on APCs outside of the antigen binding site, leading to overwhelming the release of IL-1, IL-2, interferon gamma, and TNF alpha, leading to shock. Not a good situation. Now, toxic shock syndrome, fever, rash, shock, other toxins that cause scalded skin syndrome, like exfoliative toxin does that. And food poisoning, which heat-stable enterotoxin does that. Remember that TSST1 is mainly really working on the shock effect, not so much these other kinds of things necessarily. What about strep pyogenes? Strep pyogenes, group A strep. This has something called the erythrogenic toxin, which your book calls, or erythrogenic exotoxin A, which your book just calls exotoxin A. Again, it says here, cross-links beta region of the TCR to the MHC class II on, remember, MHC means major histocompatibility complex, by the way. Class 2 on APC is outside of the antigen binding site, leading to overwhelming release of IL-1, IL-2, interferon gamma, and TNF alpha, leading to shock. Now it says here, toxic shock-like syndrome, this is for strep pyogenes, right? Leading to fever, rash, shock, and scarlet fever. Now there's this little thing called endotoxin. So endotoxin right here, it says here, LPS found in the outer membrane of gram-negative bacteria, both cocci and rods, composed of O antigen plus core polysaccharide plus lipid A, the toxic component. The cilia have lipooligosaccharide, released upon cell lysis or by living cells by blebs detaching from the outer surface membrane versus exotoxin, which is actively secreted. Three main effects would be macrophage activation, toll-like receptor 4/CD14, complement activation, and tissue factor activation. So again, the three things of endotoxin you're going to have are macrophage activation from TLR4 and CD14, complement activation, and then tissue factor activation. So you're activating macrophages, complement, and tissue factor. Endotoxins, this is the little learning device here. Edema, nitric oxide, DIC/death, outer membrane, TNF alpha, O antigen plus core polysaccharide plus lipid A. Extremely heat stable. Okay. Look, pushing in a little bit with that one. IL-1, IL-6, neutrophil, chemotaxis, and shock. There's a little diagram right here. It says endotoxin, lipid A component, it has 3 different effects: macrophage activation through TLR4 and CD14, complement activation, and tissue factor activation. So the macrophage activation leads to IL-1 and IL-6 release, TNF-alpha release, and nitric oxide. IL-1 and IL-6 causes fever. TNF-alpha will cause fever and hypotension, I should say, and nitric oxide causes hypotension. This is actually pretty good. This is a pretty good diagram because it actually helps you understand how a lot of these reactions work. You know, tells you, oh, well, this thing does this and this thing does that. Okay. Complement activation leads to C3A and C5A. C3A is involved in histamine release, hypotension, and edema. And C5A also does all that stuff, plus it also does neutrophil chemotaxis. True. We also have tissue factor activation leads to coagulation cascade, obviously, which can lead to DIC, which you do not want to have. That's a very serious problem. Now we're going to go on to something called microbiology. Now going into the clinical bacteriology section. And I'll take a break and come back to you soon. Just if you're watching this it's nothing more than a recording at that point, just skip to the forward to where I'm start talking again, okay? Okay, we're back, and we're in the clinical bacteriology section of the microbiology chapter, focusing on gram-positive lab algorithms. So this is like a way for you to figure out, okay, well, I have an organism that's gram-positive. How can I figure out what it is specifically? Well, is it a bacilli, cocci, or branching filaments? Bacilli is in like rod-shaped, cocci is more as like a circular shape, and then branching filaments, that's its own thing. Let's say it's bacilli. Is it aerobic or anaerobic? If it's aerobic, maybe it's Listeria bacillus, like Corynebacterium diphtheria. If it's anaerobic, maybe think something like Cutibacterium, which is formerly called Propinobacterium. What about if it's branching filaments? Was it aerobic or anaerobic? If it's aerobic, there'll be nocardia, which is weakly acid-fast. What about anaerobic type? That'd be actinomyces, which is not acid-fast. What about if it's cocci? Well, is it catalyzed positive or negative? If it's catalyzed positive, then it's probably the grape cluster-shaped Staphylococcus. If it's gram-negative, then it's going to be Streptococcus, which is that chain formation. So it's Staphylococcus, this sort of catalyzed positive type. It's either coag positive or coag negative. If it's coag positive, it's Staph aureus. If it's coag negative, well, it's either going to be Saprophyticus or epidermidis. Epidermidis is going to be novobiocin sensitive. Saprophyticus is going to be novobiocin resistant. I don't like how they say positive or negative. They should say sensitive or resistant, but whatever. Epidermidis is sensitive. Saprophyticus is resistant to novobiocin. What if it's Streptococcus? Well, then it has to be alpha, beta, or gamma hemolytic. Now, if it's beta hemolytic, by the way, that's important. Beta hemolytic, that's going to be complete hemolysis. I don't like how they do the naming convention on this. They say, because alpha is partial, beta is full, and then gamma is none. So the order should be beta, alpha, gamma, because group A strep is the first one, group B strep should be the second one. Those are both beta hemolytic. And then group D strep, which is in gamma hemolytic, that's going to be, the order is weird, okay? But let's say it completely hemolyzes it. It's beta hemolytic. It's either going to be bacillitrazine sensitive, group A strep, strep pyogenes, or it's going to be bacillitrazine resistant, group B strep, strep agalectiae. Now, what about if it's alpha hemolytic? So it can hemolyze somewhat, but not entirely. It's partial. Well, it's either going to be apoptosis sensitive and biosoluble, like strep pneumo, which is encapsulated, or it's going to be apoptosis resistant, and it's going to be bioinsoluble, like Viridans species. That can include strep mutans. They say strep mitis right there. Maybe strep sanguinis, I'm not quite sure. But definitely strep mutans, yeah. Those would be bioinsoluble. Those are going to be apoptosis resistant. Now, what if it's gamma? What if it doesn't hemolyze at all? It's gamma hemolytic. Well, then can it deal with salt? If it can deal with salt, then it's probably Enterococcus, like group D strep, which is E. faecium, Enterococcus faecium, Enterococcus faecalis. If it can't handle the salt, there's going to be strep bovis or strep galliticus, which can cause colon cancer and also can cause some sort of a heart issue as well. Just be aware of that, by the way. Galliticus and slash bovis, that can cause a kind of carditis. I think it might be myocarditis, but it's one that's associated with colon cancer. What kind of carditis is caused by Galliticus? Infective endocarditis. Okay, so it's endocarditis for bovis and slash Galliticus. Now let's go on to this. This is talking about gram-pot. Well, my book says here hemolytic bacteremia, or bacteria, hemolytic bacteria. So we'll go into that now. You can read this if you want to. Hemolytic bacteria. We have alpha hemolytic and beta hemolytic. So alpha hemolytic, partial oxidation of the hemoglobin leading to a greenish or brownish color without clearing around the growth on the blood agar. This includes Streptococcus pneumonia and Strep viridans species. What about beta hemolytic bacteria? complete lysis of RBCs, leading to a pale or clear area surrounding the colony on the blood agar, as you can see in the picture. Now, includes Staphylococcus aureus, Streptococcus pyogenes, group A strep. It also includes Streptococcus agalactiae, which is group B strep, and Listeria monocytogenes. So obviously it's going to include group A and group B strep, but also Listeria, also Staph aureus as well. What about Staphylococcus aureus, Staph aureus? It's a gram-positive, beta-hemolytic, catalase-positive, coagulase-positive cocci in clusters. Protein A, virulence factor, bonds the Fc-IgG, inhibiting complement activation and phagocytosis. Commonly colonizes the skin, nares, ears, axilla, and groin. Causes inflammatory disease, skin infections, organ abscesses, pneumonia, often after influenza virus infection, infective endocarditis, septic arthritis, and osteomyelitis. Toxin-mediated disease, toxic shock syndrome, TSST1, scalded skin syndrome, exfoliative toxin, and rapid onset food poisoning from enterotoxins. A lot of stuff. Very nasty. What about MRSA? MRSA is a methicillin-resistant Staph aureus. Important cause of serious healthcare-associated and community-acquired infections. Resistance due to an altered penicillin-binding proteins conferred by MEK-A gene. Some strains release pantin-valentine leukocidin, PVL, which kills leukocytes and can cause tissue necrosis. TSST1 is a superantigen that binds to the MHC class II and T cell receptor, resulting in polyclonal T cell activation and cytokine release. Staphylococcal toxic shock syndrome, TSS, causes leading to fever, vomiting, diarrhea, rash, desquamation, shock, and organ failure. TSS results in an increase in AST, an increase in ALT, and an increase in bilirubin, associated with prolonged use of vaginal tampons or nasal packing. Compared with Streptococcus pyogenes, TSS, which is, again, compared to the Strep pyogenes toxic shock syndrome, which is a toxic shock-like syndrome associated with the painful skin infection. So it's somewhat different. Staph aureus food poisoning due to an ingestion of preformed toxin, leading to a short incubation period of 2 to 6 hours, followed by a non-bloody diarrhea and emesis. Non-bloody diarrhea and emesis. Enterotoxin is heat stable, not destroyed by cooking. Staph aureus makes coagulase and toxins, forms fibrin clot around itself, abscess. Okay. So these are good. This is actually good stuff because I have my own little slides for this kind of stuff to review. Oh, what is Staph aureus? What is this like? So forth. This is actually a pretty good, concise. It has as much detail as you might need. Obviously there's a lot more information that's relevant for Staph aureus than let's say Staph epidermidis, and so they have more information on it. So I think that's pretty good. Staph epidermidis, gram-positive, catalase positive, coag negative, urease positive cocci in clusters. Novobiocin sensitive. It does not ferment mannitol versus Staph aureus. Normal microbiota of skin, contaminates blood cultures. Infects prosthetic devices like hip implant, heart valve, IV catheters by producing adherent biofilms. Okay. What about Staph saprophyticus? Again, remember, Staph epidermidis and saprophyticus are Staph species that are coag negative, unlike aureus, which is coag positive. Let's go back to Staph saprophyticus now. It's a gram-positive, catalase positive, coag negative, urease positive, cocci in clusters, novobiocin resistant, normal microbiota of female genital tract and perineum. So it's actually, it is actually part of the normal flora. Normal microbiota of the female genital tract and perineum. Not necessarily inherently pathological if it's found there. It is the second most common cause of UTIs in young females. Most commonly is E. coli. So remember, E. coli is number 1 for UTIs, bacterial UTIs, but number 2 is saprophyticus. So just be aware of that. That is an important detail. Now I got something called strep pneumo. Strep pneumo is a gram-positive alpha-hemolytic lancet-shaped diplococci, as you can see in the picture. It is encapsulated IgA protease, aptogen sensitive, and biosoluble. Most commonly cause causes MOPS, meningitis, otitis media in children, pneumonia, and sinusitis. Pneumococcal pneumonia is associated with rusty sputum. Patients with anatomic or functional hyposplenia or asplenia are predisposed to infection. No virulence without capsule. Pneumococcal vaccines are available in both conjugate PCV13, PCV15, and PCV20, and polysaccharide PPSV23 formulations. Okay. Not entirely sure what that exactly means, or if I even need to know that for the board exam, but I'll have to look into it if I need to. Now, Viridans group streptococci. Viridans group streptococci. Now, of course, I know the MOPS stuff. Yeah, MOPS, meningitis, otitis media, pneumonia, and sinusitis. I know that. I'm aware of that information. The vaccine stuff, I'm not as familiar with that kind of stuff. Is that necessary to know? Well, they put it in the new book, so they think it might be useful. So now let's go back to Viridans group streptococci. So strep viridans, which includes, I think, strep mutans as well. So strep viridans, gram-positive alpha hemolytic cocci, apoptosin-resistant and bioinsoluble, normal microbiota of the oral pharynx, Streptococcus mutans and Streptococcus mitis cause dental caries. True. Streptococcus sanguinis, so they're all part of the same group. Streptococcus sanguinis makes dextrans that bind to fibrin platelet aggregates on damaged heart valves, causing infective endocarditis. Remember, the dextrans are part of the biofilm. So when you hear dextrans, think biofilm for strep viridans. Viridans, those two things are connected. Viridans group strep live in the mouth because they are not afraid of the chin, optochin, resistant. Okay, sure. Sanguinis equals blood. Yes, sanguine, right? Blood. Think there's a lot of blood in the heart, infective endocarditis. Okay, that one's not too bad. As far as learning devices go. Strep pyogenes, group A, streptococcus, finally. This is beta hemolytic now. It's gram-positive cocci and chains, as you can see in the picture. Group A strep causes pyogenic, pharyngitis, cellulitis, and impetigo, honeycrusted sputum, honeycrusted lesions, Erypselis, I'm just thinking from memory. Erypselis. It's toxigenic, scarlet fever, toxic shock-like syndrome, necrotizing fasciitis, that's important. Immunologic, rheumatic fever, glomerulonephritis. Bacitracin sensitive, beta hemolytic, pyrrolidonyl, arolamidase, PYR positive, hyaluronic acid capsule, and M protein inhibits phagocytosis. Antibodies to M protein enhance host defenses. Structurally similar to host proteins like myosin. It can lead to autoimmunity, carditis seen in acute rheumatic fever. Diagnose strep pharyngitis via throat swab, which can be tested with an antigen detection assay, rapid in-office results, or cultured on blood agar. Results in 48 hours. And they don't have Jones criteria in this one. I'm surprised by that. Jones criteria, I like that. Now, my book says this. I'll say what my book says first, and I'll read what you have on your little thing there. It says pH, pyogenes, pharyngitis can result in rheumatic fever and glomerulonephritis. Strains causing impetigo can induce glomerulonephritis. Key virulence factors include DNase, erythogenic exotoxin, tryptokinase, streptolysin O, ASO titer, or anti-DNase B indicates recent Streptococcus pyogenes infection. Scarlet fever, fine, blanching, generalized sandpaper-like rash, sparing palms and soles, strawberry tongue, and circumoral pallor in the setting of a group A strep pharyngitis. Erythogenic toxin A positive. Now your book has this thing called the Jones criteria, which I get. Maybe they say, "Oh, it's not perfect, isn't the best." Maybe it's an opinion basically. What do you think is good, or what do you think is a better learning tool or not? I like it. I was grown on me over time. So Jones criteria, major criteria for the acute rheumatic fever. So J for joints, polyarthritis. The O is really a heart, and the heart means carditis. The N is nodules, subcutaneous nodules. The E is for erythema marginatum. And the S is for Sydenham chorea. So just keep that in mind. because the scarlet fever is a pretty rheumatic fever. Scarlet fever, you should be aware of that as well. But rheumatic fever is an important part of strep pyogenes, so just be aware of that. What about strep agalactiae? Not as important, I have to admit. Not as important. You should know it, but not nearly as important as group A strep. So Streptococcus agalactiae, group B streptococci. Gram-positive cocci, bacillus resistant, beta hemolytic, colonizes the vagina, causes pneumonia, meningitis, and sepsis, mainly in babies. Polysaccharide capsule confers virulence, produces the CAMP factor. That's not cyclic AMP, it's called CAMP factor. CAMP factor with a big C, AMP. CAMP factor, which enlarges the area of the hemolysis formed by Staph aureus. Note, CAMP stands for the author of the test, not cyclic AMP. Thank you for saying what I said before you got to it. Okay. Heparate test positive, PYR negative, which is, again, PYR in this context means the pyrrolinodyl aryl limidase, which is PYR positive for strep pyogenes, which is group A strep. So group A strep is PYR positive, but group B strep is PYR negative. Anyway, screening pregnant patients at 35 to 37 weeks of gestation. We're still talking about by agalactiae now. For agalactiae, screen pregnant patients at 35 to 37 weeks of gestation with rectal and vaginal swabs. Patients with a positive culture receive intrapartum penicillin or ampicillin prophylaxis. Group B for babies. Okay, that one's not too bad. Not convoluted or anything. Streptococcus bovis. Formerly called strep bovis. They say strep. My book says strep galolyticus, but your book says strep bovis. It's the same thing. Strep galolyticus, formerly called strep bovis, before it was, changed at some point. Gram-positive cocci, colonizes the gut, can cause bacteremia and infective endocarditis. Patients with strep galolyticus endocarditis have an increased incidence of colon cancer. Bovis in the blood, cancer in the colon. Okay, that's, if you want to remember it that way, you can do that. That's fine. Enterococci. And remember, enterococci is not really even, his name is a little bit different. It's called group D strep historically. Anyway, enterococci, gram-positive cocci, enterococci, including Enterococcus faecalis and Enterococcus faecium, are normal colonic microbiota that are intrinsically resistant to penicillin G and cause UTI, biliary tract infections, and infective endocarditis following the GI-GU procedures. Catalase negative, PYR positive, also PYR positive, right? Typically non-hemolytic. Yeah, it's normally gamma hemolytic. VRE, vancomycin-resistant enterococci, are an important cause of healthcare-associated infection. Enterococci are more resistant than streptococci, and they can grow in 6.5% salt, sodium chloride, and bile lab test. They're very hardy. Entero means intestine. Entero, like inside your enteric system. Entero. Intestine. Faecalis means feces. Strepto means twisted chains. And coccus means berry. Yeah, when we talk about streptococcus, we're talking about chains of little berries. That's why it looks like that. That's the description of it. Make sense? They're chains of little berries in shape, and they're inside your intestines, in the case of Enterococcus. Now, Bacillus anthracis. Let's talk about it. Bacillus anthracis, which is a gram-positive spore-forming rod that produces anthrax toxin, exotoxins consisting of protective antigen lethal factor, which inhibits the MAP kinase. That's, again, lethal factor is the other toxin. We're talking about edema factor before. Now we're talking about the lethal factor. Lethal factor inhibits the MAP kinase, leading to macrophage apoptosis. And edema factor, which acts as an adenyl cyclase, leading to increase in intracellular cyclic AMP, upsetting the homeostasis, leading to edema and necrosis. It has a polypeptide capsule, poly-D-glutamate. Colonies show a halo of projections, sometimes called Medusa head appearance. Now, cutaneous anthrax, painless papule surrounded by vesicles, leading to an ulcer with black eschar. Painless necrotic, uncommonly progresses to bacteremia and death. Pulmonary anthrax, the inhalation of spores, most commonly from contaminated animals or animal products, although also a potential bioweapon, leading to flu-like symptoms that rapidly progress to fever, pulmonary hemorrhage, mediastinitis, chest x-ray may show widened mediastinum, and shock. Also called wool sorter's disease, prophylaxis with ciprofloxacin or doxycycline when exposed, Both both cutaneous and pulmonary anthrax may be complicated by hemorrhagic meningitis. Yeah, okay. So the presentation looks different depending on whether that's the cutaneous type or the pulmonary type, which makes sense because there's different places it's attacking from. What about Bacillus cereus? Bacillus cereus, gram-positive rod, causes food poisoning. Spores survive cooking rice, reheated rice syndrome. Keeping rice warm results in germination of spores and enterotoxin formation. Emetic type causes nausea and vomiting within 1 to 5 hours, caused by cerulide, a preformed toxin. Diarrhea types cause watery, non-bloody diarrhea and GI pain within 8 to 18 hours. Management would be supportive care. Antibiotics are ineffective against toxins. Okay. Yeah, because it's a toxin. Even if it's dead, the toxin is still there, so. Okay. I'm going to take a brief break. Anyway, Clostridium with exotoxins. Clostridioides difficile produces toxins A and B, which damages enterocytes. Both toxins lead to watery diarrhea, leading to pseudomembranous colitis, often secondary to antibiotic use, especially clindamycin, ampicillin, cephalosporins, fluoroquinolones associated with PPIs. Fulminant infection, toxic megacolon ileus shock. Difficile causes diarrhea. Diagnosed by PCR or antigen detection of one or both toxins in stool. Treatment: oral vancomycin or fidaxomicin. For recurrent cases, consider repeating prior regimen or fecal microbiota transplant. What about Clostridia? So Clostridia species, we're talking about Clostridioides difficile. What about Clostridium? It says here Clostridium, gram-positive, spore-forming, obligate anaerobic rods. Tetanus toxin and botulinum toxin are proteases that cleave snare protein involved in neurotransmission. So what about Clostridium tetani? C. tetani. Pathogen is non-invasive and remains localized to wound site. Produces a tetanospasmin, which is an exotoxin causing tetanus. Tetanospasmin is spread by retrograde axonal transport to the CNS and blocks release of GABA and glycine from Renshaw cells in the spinal cord. Now, this causes a spastic, i.e. tetanic paralysis, trismus, which is lockjaw, rhasis, R-I-S-U-S, sardonicus, raised eyebrows and open grin, and opisthotonos, spasms of spinal extensors. Now, tetanus is tetanic paralysis, hence the name. Prevents with tetanus vaccine. Treat with antitoxin plus or minus vaccine booster. Antibiotics, diazepam for muscle spasms, and wound debridement. Okay. Where was I? Okay, sorry. And wound debridement. Yeah. That seems a little bit harsh. Wound debridement seems pretty serious. What about botulinum? We were talking about that before. Botulinum. C. botulinum. Produces a heat labile toxin that damages the snare proteins, thus preventing the acetylcholine release at the neuromuscular junction, causing botulism. In babies, ingestion of spores, e.g. in honey, leads to disease, floppy baby syndrome. In adults, disease is caused by ingestion of preformed toxin, like in canned food. Symptoms of botulism, the 5 Ds: diplopia, which is double vision, dysarthria, dysphagia, dyspnea, and descending flaccid paralysis. It does not present with sensory deficits. Botulinum is from bad bottles of food, juice, and honey. Sure, that's the learning device. And it says your treatment, human botulinum immunoglobulin is a treatment for the toxin. Local botulinum toxin A, Botox injections are used to treat facial dystonia, hyperhidrosis, and muscle spasms, and cosmetic reduction of facial wrinkles. So they figured out how to use the toxin for cosmetic purposes, etc. What about Clostridium perfringens? So this C. perfringens produces alpha toxin, lecithinase A phospholipase, that can cause myonecrosis, which is gas gangrene. It causes gangrene. It presents as a soft tissue crepitus and hemolysis. If heavy spore-contaminated food is cooked but left standing too long at less than 60 degrees Celsius, spores germinate, leading to vegetative bacteria ingestion and leading to enterotoxin, which leads to a late onset, 10 to 12 hours, food poisoning symptoms, resolution in 24 hours. Okay, so I see. Perfringens perforates a gangrenous leg, spontaneous gas gangrene via hematogenous seeding, associated with colonic malignancy, is most commonly caused by Clostridium septicum. Okay. And I've heard before that you might want to use debridement in certain cases as well for gangrene. If you have gangrene, which is not fun, never been in that situation, thankfully, but it's not something you want to have. You might need to use debridement. You might need to remove the diseased necrotic tissue. And you can see a picture right there for perfringens. It does not look fun. What about C. What about my book had C. difficile. I mentioned it before already. I don't need to go through that twice. So you can read about that right there. That's fun. Let's go back to Corynebacterium diphtheria. Corynebacterium diphtheria is a gram-positive rod occurring in angular arrangements, transmitted via respiratory droplets, causes diphtheria via exotoxin encoded by beta prophage. Potent exotoxin inhibits the protein synthesis via the ADP ribosylation of EF2, leading to possible necrosis in pharynx, cardiac, and CNS tissue. Symptoms include pseudomembrane pharyngitis, grayish-white membrane with lymphadenopathy, bull's neck appearance. Toxin dissemination may cause myocarditis, arrhythmias, and neuropathies. Lab diagnosis based on gram-positive rods of metachromatic blue and red granules and positive ELEC test for toxin. Toxoid vaccine prevents diphtheria. Coryn right there means club-shaped. Metachromatic granules of Loeffler media. It says black colonies on cysteine tellurite agar. This is the learning device ABCDEFG, ADP ribosylation, beta prophage, Corynebacterium, diphtheria, elongation factor 2, by the way, inhibition, and granules. Treatment would be diphtheria, antitoxin plus or minus erythromycin or penicillin. So the antitoxin is relevant because the problem is the toxin primarily, but, the toxin is what you're immediately sensing is the issue afoot is the actual toxin itself, but obviously you don't want to have the bacteria that's causing the toxin to be around either. So that's why you would have plus or minus the penicillin or erythromycin. What about Listeria monocytogenes? Gram-positive facultative intracellular rod acquired by ingestion of unpasteurized dairy products and cold deli meats, transplacental transmission, and by vaginal transmission during birth. Grows well at refrigeration temperatures, cold enrichment. Forms rocket tails, red in the picture, via actin polymerization that allows intracellular movements in cell-to-cell spread across cell membranes, thereby avoiding antibody. Listerolysine O generates pores in phagosomes, allowing its escape into cytoplasm. Characteristic tumbling motility in broth can cause amnionitis, sepsis, and spontaneous abortion in pregnant patients, granulomatosis, infantiseptica, meningitis in immunocompromised patients, neonates, and older adults, mild self-limited gastroenteritis in healthy individuals. Treatment would be ampicillin. Nocardia versus actinomyces. Both are gram-positive and form long, branching filaments resembling fungi. We have Nocardia and we have actinomyces. So Nocardia species is aerobe, catalase-positive, acid-fast, weakly acid-fast, found in the soil, causes pulmonary infections in immunocompromised, can mimic TB but with a negative PPD test, Cutaneous infections after trauma in immunocompetent people can cause spread to CNS, which leads to cerebral abscess. Not fun. Treatment would be with sulfonamide like Bactrim, which is trimethoprim-sulfamethoxazole. And you could treat... Okay, and I'll read you that later. Treatment is a SNAP, sulfonamides, nocardia, actinomyces, penicillin. Okay. Actinomyces is an anaerobic, catalase negative organism that's not acid-fast. It's normal oral, reproductive, and GI microbiota. It causes oral/facial abscesses that drain through the sinus tracts, often associated with dental caries, extraction, and other maxillofacial trauma. Okay. Forms yellow sulfur granules and can cause PID and IUDs. Okay, fair enough. You treat with penicillin. And again, the treatment would be this. So treatment is a SNAP, sulfonamides for nocardia, like Bactrim, and actinomyces with penicillin. My book goes into mycobacteria. Let me see if I can find that in here. Right here. Okay. That was easy enough. Is it TB up? Is TB up here? Yeah, TB is right there. TB. So let's talk about mycobacteria first. Mycobacterium is a acid-fast rods, pink rods, arrows shown in picture A. But my book's a little bit different. Your thing says all mycobacteria are acid-fast organisms, pink rods and arrows in A. Okay. Acid-fast rods grow slowly in culture. Mycobacterium tuberculosis, TB, often resistant to multiple drugs. Mycobacterium avium intracellulare causes disseminated non-TB diseases and AIDS, often resistant to multiple drugs. Mycobacterium scrofulaceum, cervical lymphadenitis in children, and the Mycobacterium marinum, skin infection in aquarium handlers. Yes, that's true. TB symptoms include fever, night sweats, weight loss, cough, non-productive or productive, hemoptysis. Cord factor creates a serpentine cord appearance in virulent M. tuberculosis strains, activate the macrophages promoting granuloma formation, and induces release of the TNF-alpha. Sulfatides, surface glycolipids, inhibit phagolysosomal fusion. So when they ask you, for example, what element of the mycobacteria prevents it from being broken down via the phagolysosomal fusion, what's preventing phagolysosomal fusion? The answer is the sulfatides. When they ask about the serpentine cord, think about cord factor. And that promotes the granuloma formation. So what about TB? What about tuberculosis? Tuberculosis is PPD positive if current or past infection, is PPD negative if there's no infection and in immunocompromised patients, especially with low CD4 positive cell count. So even if you actually have TB ongoing, if you have such a poor immune response because you're immunocompromised, you still might be considered PPD negative because PPD is operating under the context of an immune reaction. And so you will not have an immune reaction even if you have TB, if you are severely immunocompromised. So just keep that in mind. Context is key. Interferon gamma release assay, IGRA, has fewer false positives from BCG vaccination. Caseating granulomas with central necrosis and Langhans giant cells, single example in picture A. They're saying my book says that's in Langhans giant cells for A. Your book says that's a caseating granuloma with central necrosis. Whatever. Caseating granulomas with central necrosis and Langhans giant cell. Okay. Caseating granulomas with central necrosis and Langhans giant cell. Okay. Whatever. It is what it is. Are characteristic of secondary tuberculosis. Do not confuse Langhans cells, which are fused macrophages, with Langerhans cells, which are dermal APC. So Langhans cells are like giant cells that are fused together that are macrophages. Langerhans cells, completely unrelated thing. Probably a different guy, I would assume, right? And that's related to the dermal APC. So dermal antigen presenting cells, those are Langerhans cells. These giant multicellular, they fused cell macrophages cells related to TB, that's called a Langerhans cell. Now, TB activation risk is highest in immunocompromised individuals, for example, HIV, organ transplant recipients, TNF-alpha inhibitor use, Reactivation has a predilection for the apices of the lung due to the bacteria being highly aerobic. So because you have, because they're highly aerobic, they prefer the upper lobes, basically, the apices of the lung. Let's talk about leprosy, which your book calls Hansen disease. Leprosy has been around for quite a while. So leprosy, also called Hansen disease, is caused by Mycobacterium leprae, an acid-fast bacillus that likes cool temperatures, infects skin and superficial nerves, glove and stocking, loss of sensation, and cannot be grown in vitro. Diagnosed via skin biopsy or tissue PCR, reservoir in the United States is armadillos. Leprosy has two forms. Many cause cases fall temporarily between the two extremes. Lepromatous leprosy presents diffusely over the skin with leonine, lion-like facies, shown in the picture in B. It is communicable, high bacterial load, characterized by low cell-mediated immunity with a largely Th2 response. Lepromatous form can be lethal. The tuberculoid form, limited to a few hypoaesthetic hairless skin plaques, characterized by high cell-mediated immunity with a largely Th1 response and low bacterial load. So basically, the tuberculoid form is the less severe form. It means you have a much stronger immune response that can handle it a lot better. The lepromatous form, where you're really messed up looking, that's more of an issue where the immune system really can't handle the organism as well, and as a consequence, you get much more severe presentation. Now, here's the thing: treatment: dapsone and rifampin for tuberculoid form, and clofazimine is added for lepromatous form. Daring rifles target cloaked leprechauns. Okay, whatever. Just know that rifampin and dapsone are the ones you want to use in all cases, but you want to add clofazimine on top of that if you have the more severe lepromatous form. And this is a little chart right here for gram-negative algorithm. Okay, let's go back to the gram-negative algorithm. So let's say we have a gram-negative organism, right? We want to figure out, like, what's going on with this? What do we think it is? Well, let's think about it. Let's say, for example, we think it's diplococci. What could it be? What if it's diplococci, it's aerobic, and we have to ask basically, it's basically going to be Neisseria, with an exception to that rule, but Neisseria, right? We're going to say, okay, is it maltose fermenting or maltose non-fermenting? If it's fermenting, then it's meningitidis. If it's not fermenting, then it's Neisseria gonorrhea. They also say Moraxella. Moraxella, sure. I would not care as much about Moraxella. You should know what Moraxella is. Fair enough. Actually, no. You know what? You should know that because I've seen that on exams before. Moraxella. It's not as big of a topic for studying, typically speaking, but you should know what Moraxella is. In all fairness. But Neisseria species, the fermenting type that can ferment maltose is the meningitidis. The one that cannot is gonorrhea. Gonorrhea is the one that's the STD. Or the STI, Anyway, Coccobacilli is the next category. Coccobacilli, this is a little grab bag, basically. We have Haemophilus influenza, Bordetella pertussis, Pastorella, Brucella, Francisella tularensis, and Actinobacter baumannii. Okay, so... And they say in your book here, H. flu needs factors 5 and factor 10. True. Now, the way I remember that category of Coccobacilli is simply try to do this little sort of what's the correct term for that? When they have a certain kind of sound to them every single time. I say, influenza, Pastorella, Brucella, Francisella, Bordetella, that kind of thing. Because they all sound like that. And Yersinia should be in there as well. Yersinia, Francisella, Brucella, Pastorella, Bordetella, influenza. Yersinia should be in there, but they don't mention it here, I don't think. Anyway, next one's curved rod types. They have 3 down here. I usually think of 2, but they have 3. These are oxidase positive as well. We have Campylobacter jejuni, which grows in 42 degrees Celsius. We have Vibrio cholera, which just grows in alkaline media. And they also have here is produces urease, Helicobacter pylori. Now, I think about Helicobacter pylori in the same camp as I think about, let me think. Is it H. pylori? I think about H. pylori along with, I think, maybe Pseudomonas or something like that. No, I might be confusing that now. Give me a moment, actually, quickly. Let me see if I have it in my notes somewhere. Oh, does this work? Here we go. I have my own notes on how I do categorization. And let me see if I have it on hand. So my notes, the way I do it, this is from the Boards and Beyond videos, which you can go watch if you want to. If you watch Boards and Beyond, you can do that. I usually categorize H. pylori with Pseudomonas as a rod that is lactose non-fermenting and is oxidase positive. Now they say, oh, this is comma-shaped. It's comma-shaped. I don't know. Is H. pylori more comma-shaped or is it more rod-shaped? I don't know. But H. pylori, Pseudomonas aeruginosa is what I was thinking. Yeah, Pseudomonas. Okay. So, pick your poison. Whatever you prefer, whatever your organization scheme you prefer, you can go for that, I guess. Just make sure it's correct. What about bacilli? Bacilli we have lactose fermentation. We talked about that before. These are rods, right? Can they ferment it or can they not ferment? If they can ferment, is it slow? Well, in that case, it's Citrobacter and Serratia. Remember, slow Citrobacter, slow Serratia. What about fast? We'll think about Klebsiella, E. coli, and Enterobacter. And what about the ones that are oxidase? Well, sorry, that cannot ferment. They cannot ferment lactose. They're either oxidase positive or negative. If it's oxidase positive, they say Pseudomonas. Again, arguably Helicobacter falls into that category along with it. But they say, no, that's definitely comma-shaped. I'm not sure about that, but whatever. Pseudomonas is oxidase positive. The ones that are oxidase negative, they divide further between, it says here, H2S production on TSA agar, they say Shigella or Yersinia, in one category are negative, and the ones that are positive are Salmonella and Proteus. Again, I would say Yersinia is a coccobacilli and should not be in that category, but again, I guess it's debatable. Whatever. And they say here the ones that are bold, the ones you should actually care about, important tests are in bold, important pathogens are in bold italics. Okay, fair enough. And with that being said, just because they say, oh, well, these ones are bold, they're more important. Yeah, I mean, that's kind of true. My book puts Moraxella, by the way, as being not bold, but I can assure you I have had exams where Moraxella is mentioned. And so you should probably know what that is, because you might get one question on the exam. Oh, one question. But it's better to know that than not know it, I guess. Okay, now we're on to the next section. We're talking about Neisseria now. Neisseria, and by the way, we're on page 140. We're wrapping up, I think, on 152 or something like that. Yeah, 152, which means we're wrapping up really on 151. So we have, like 11 or so pages, basically. So Neisseria. Neisseria, here we go. Neisseria is a gram-negative diplococci, metabolizes glucose and produces IgA proteases, contains lipooligosaccharides with strong endotoxin activity. Neisseria gonorrhea is often intracellular within neutrophils, which is shown in my picture. Your book doesn't have it. Acid production, meningococci, maltose and glucose, gonococci, glucose. So they're saying this is like a little learning device. Acid production, meningococci is maltose and glucose, but gonococci are glucose. So gonococci versus meningococci. I'd assume the difference between these two different things are referring to when they say meningococci, what they really mean is Neisseria meningitidis, And when they say gonococci, they probably mean Neisseria gonorrhea, would be my intuition. So gonococci, i.e., I'd assume gonorrhea. It has no polysaccharide capsule, true. No maltose acid detection, true. No vaccine due to antigenic variation of pilus proteins. It's sexually or perinatally transmitted. It causes gonorrhea, septic arthritis, neonatal conjunctivitis 2 to 5 days after birth, pelvic inflammatory disease, PID, and Fitzhugh-Curtis syndrome, which is related to that. If I'm not mistaken, I think it's connected to PID. Now, diagnosed with NAAT, condoms decrease sexual transmission. Not having sex will also do that too. Actually, a little bit better than that. Actually, you just don't do it. You know. Erythromycin eye ointment prevents neonatal blindness. Treatment: single dose of IM ceftriaxone. If chlamydia co-infection not excluded by molecular testing, add doxycycline. What about meningococci? Well, it has a polysaccharide capsule. It is encapsulated. It's maltose acid detection. It is ferment maltose. There is a vaccine for meningitis vaccine, type B vaccine, available for at-risk individuals. Transmitted via respiratory and oral secretions, more common among individuals in close quarters, like army barracks or college dorms. Causes meningococcemia with petechial hemorrhages and gangrene of toes. Meningitis, Waterhouse-Friderichsen syndrome, very important, which is an acute hemorrhagic adrenal insufficiency. Okay? Diagnosed by the culture-based test or PCR, you treat it with rifampin, ciprofloxacin, or ceftriaxone prophylaxis in close contacts. And then you also have treatment, ceftriaxone or penicillin G. Now what about Haemophilus influenza? Haemophilus influenza, small gram-negative coccobacillary rod transmitted through respiratory droplets, non-typable unencapsulated strains are the most common cause of mucosal infections, otitis media, conjunctivitis, bronchitis, as well as in invasive infections since the vaccine for the capsular type B was introduced. Produces IgA protease, culture on the chocolate agar, which contains factors 5, which is NAD+, and factor 10, which is hematin, for growth. Remember, hematin is the iron factor. It can also be grown with Staph aureus, which provides factor 5 via the RBC hemolysis. Oh, interesting. Hemophilus causes epiglottitis. Endoscopic appearance can be cherry red in children. Thumb sign on the lateral neck X-ray, which is shown in the picture B for you. Meningitis, otitis media, and pneumonia. Okay. So, vaccine contains type B capsular polysaccharide, which is the polyribosyl ribitol phosphate, conjugated to diphtheria toxoid or other protein, given between 2 and 18 months of age. It does not cause the flu. Influenza virus does. Treatment would be amoxicillin plus or minus clavulanate for the mucosal infection, ceftriaxone for meningitis, rifampin prophylaxis for close contacts. Bucaldaria. My book has something on Bucaldaria cepacia complex, which I don't know if your book does or not. If it does not, I will still say it, but it looks like they might skip it. My book goes from Bucaldaria to the Bordetella, so I'll just do the Bucaldaria cepacia complex thing. So Bucaldaria cepacia complex, aerobic catalase positive gram-negative rod, causes pneumonia in patients with underlying lung disease, such as cystic fibrosis, often multi-drug resistant. Infection is a relative contraindication to undergoing lung transplant due to its association with poor outcomes. Bordetella pertussis, gram-negative Arabicoccobacillus. Virulence factors include pertussis toxin, which disables G sub I, adenylate cyclase, a toxin, which increases cyclic AMP, and the tracheal cytotoxin, which destroys the respiratory ciliated epithelium. 3 clinical stages: catarrhal, low-grade fever, coryza; paroxysmal, paroxysms of intense cough followed by an inspiratory whooping cough, post-tussive vomiting; and then the third, which is convalescent, gradual recovery of chronic cough. Prevented by Tdap, DTaP vaccines, Tdap for preteens and adults, DTaP for children. Produces lymphocytosis, unlike most acute bacterial infections. Treatment would be macrolides if allergic, use Bactrim, i.e. TMP, SMX, i.e. trimethoprimsulfamethoxazole. We also want brucella, which your book does not mention, but I'll talk about brucella right now. Brucella is a gram-negative aerobic coccobacillus transmitted via ingestion of the contaminated animal products like unpasteurized milk, survives in macrophages in the reticuloendothelial system, can form non-caseating granulomas, typically presents with undulant fever, night sweats, and arthralgia. Treatment would be doxycycline plus rifampin or streptomycin. Legionella pneumophila. So Legionella pneumophila, gram-negative rod, gram stains poorly, uses silver stain, grow on charcoal yeast extract medium with iron and cysteine, detected by presence of antigen in the urine. Labs may show hypoproteinemia and elevated transaminases. Aerosol transmission from environmental water source, for example, air conditioning systems, hot water tanks. Outbreaks associated with cruise ships, nursing homes. No person-to-person transmission, though. Treatment would be macrolide or quinolone. Think of a French legionnaire soldier with his silver helmet sitting around a campfire charcoal with his iron dagger. He is missing his sister, Sistine. Okay, sure. I would imagine he would have a silver medal, not a silver helmet, but whatever. Legionnaire's disease, severe pneumonia, often unilateral lobar, fever, GI, and CNS symptoms. Risk factors include older age, tobacco smoking, chronic lung disease, Pontiac fever, which is kind of connected, mild flu-like symptoms. So there's Legionnaire's disease and there's Pontiac fever. And these are caused by Legionella pneumophila. What about Pseudomonas aeruginosa? Pseudomonas aeruginosa. I've been pronouncing it wrong this whole time. Well, aeruginosa. So Pseudomonas aeruginosa, aerobic motile calyx positive gram-negative rod, non-lactose fermenting, oxidase positive, frequently found in water, increased virulence in acidic environments, has a grape-like odor. Pseudomonas is associated with pneumonia, sepsis, ecthyma gangrenosum, UTIs, diabetes, osteomyelitis, mucoid polysaccharide capsule, otitis externa, Swimmer's nosocomial healthcare-associated infections, example catheters, equipment. Addiction, injection drug use, skin infections, hot tub folliculitis, wound infection, burn victims. Mucoid polysaccharide capsule may contribute to chronic pneumonia in patients with cystic fibrosis due to viral film formation. Produces PEEP, phospholipase C, degrades cell membranes, endotoxin, fever shock, exotoxin A, inactivates EF2, green pigment A. Corneal ulcers such keratitis in contact lens wearers, minor eye trauma. Ecthyma gangrenosum, rapidly progressive necrotic cutaneous lesion caused by an Pseudomonas bacteremia, typically seen in immunocompromised patients. Treatments would be anti-pseudomonal penicillins in combination with beta-lactamase inhibitors like piperacillin, tazobactam. Third and fourth generation cephalosporins like ceftazidime and cefepime. Monobactams, fluoroquinolones, and carbapenems. Despite anti-pseudomonal activity, aminoglycoside monotherapy is avoided due to the poor performance in acidic environments. Okay, now we'll be going into Salmonella versus Shigella, but I'm going to take a break. So again, if you're watching a recording of this, just skip forward if you need to. Okay, I'm back. And yeah, I'll have to go with Salmonella and Shigella. So we're on page 142. We'll wrap up on 151. So it'll probably be about another hour to get through those 10 pages or so. Looks like I spent 15 minutes or 16 minutes or so on break. Okay, we'll see what we can do. Salmonella versus Shigella. Both Salmonella and Shigella are gram-negative rods, non-lactose fermenters, oxidase negative, and can invade the GI tract via the M cells of pyre patches. So Salmonella typhi, Salmonella species except typhi, and then Shigella. So let's talk about typhi. It says here in little parentheses, Ty-V. Sorry, reservoir spread, etc. So humans are the only reservoir for Salmonella typhi. It is spread hematogenously. It does produce H2S, which is H2S. I'm going to make sure I'm not saying this incorrectly. H2S is hydrogen sulfide. So it produces hydrogen sulfide. It has a flagella. Here's a mnemonic here: salmon swim. Okay. Virulence factors include endotoxin, Vi capsule, pronounced Ty-V. Infectious dose, ID50 would be high, large inoculum required, acid labile inactivated by gastric acids. The effect of antibiotics on fecal excretion would be prolonged duration. The immune response, primarily monocytes. Its GI manifestations, constipation followed by diarrhea. Vaccine would be oral vaccine contains the attenuated S. typhi. IM vaccine contains the Vi capsular polysaccharide. Okay. And its unique properties for Salmonella typhi causes typhoid fever, salmon-colored truncal macular rash, abdominal pain, fever, pulse, temperatures, dissociation, later GI ulceration and hemorrhage. Treat with ceftriaxone or fluoroquinolone. Carrier state with gallbladder colonization. Okay, but what about the other species, the other types of Salmonella species? So Salmonella species except typhi. Its reservoirs are humans and animals. It's spread hematogenously. Hematogenic spread is rare, so it's not usually hematogenous. It can be, but not necessarily. It is produce hydrogen sulfide? It does produce hydrogen sulfide. It does have a flagella. Its endotoxin, its virulence factor is the endotoxin. Its infectious dose is also high. The antibiotics on fecal excretion, prolonged duration. Polymorphonuclear cells, which I think are just neutrophils. In disseminated disease. Its GI manifestations include diarrhea, causes possibly bloody diarrhea. There's no vaccine for it. Only the typhi form has a vaccine. So yes, only the typhi form has the vaccine. The unique properties of the non-typhi forms is poultry, eggs, pets, and turtles are common sources of it. Treatment is supportive. Antibiotics are not indicated in immunocompetent individuals. So what about Shigella? What about Shigella? Well, the reservoir is also humans only. It's spread cell to cell, no hematogenic spread whatsoever. There's no hydrogen sulfide production. There's no flagella. The virulence factors are endotoxin and shigatoxin, which is an enterotoxin. Its infectious dose is low. Very small inoculum is required for the infection. Acid stable, resistant to gastric acids. Okay. The effects on the antibiotics on fecal excretion would shortens duration, shortens with Shigella, while for the Salmonella it prolongs it. Immune response is going to be primarily PMN infiltration, so neutrophils. GI manifestations, crampy abdominal pain leading to tenesmus, bloody mucoid stools, bacillary dysentery. There is no vaccine for it. And for some unique properties, it says the 4 Fs: fingers, flies, food, feces. In order of decreasing severity, less toxin produced. S. dysenteriae, S. flexneriae. These are Shigella species. Dysenteriae, flexneri, boydii, solnae. Invasion of M cells is key to the pathogenicity, which is in the Peyer's patches, I believe. Infectious dose is low, yeah. And M cells are the Peyer patches. Okay, so next one is Yersinia species, is Yersinia enterocolitica. So enterocolitica, gram-negative pleomorphic rod slash coccobacillus. Okay, so it's kind of debatable. They say it's a rod or it's coccobacillus. Okay, sure. So Yersinia species is Yersinia enterocolitica is a gram-negative pleomorphic rod slash coccobacillus with bipolar staining, usually transmitted from pet feces like cats or dogs, contaminated milk, or pork. Can cause acute bloody diarrhea, pseudoappendicitis, the right lower abdominal pain due to the mesenteric adenitis and or terminal ileitis, reactive arthritis in the adults. What about lactose fermenting enteric bacteria? Do we have this in here? No, we don't. What do we have? E. coli? Okay, let's talk about lactose fermenting enteric bacteria. Fermentation of lactose leads to pink colonies of MacConkey agar. Examples include Citrobacter, E. coli, Enterobacter, Klebsiella serratia. MacConkey seeks milk with seeks with an SEEKS for the different species: Citrobacter, E. coli, Enterobacter, Klebsiella serratia. EMB agar, lactose fermenter, EMB agar is a sulfur lactose fermenting enteric bacteria. EMB agar lactose fermenters grow as purple/black colonies. E. coli grows colonies with a green sheen. So, as we speak about E. coli is Escherichia coli is a gram-negative indole-positive rod. E. coli virulence factors include fimbriae, i.e. pili, P. pili, cystitis, and pyelonephritis, K. capsule pneumonia, neonatal meningitis, and the LPS endotoxin, which causes septic shock. What about strain, the toxic mechanism, and the presentation? So let's talk about the different kinds of strains. They have here entero-invasive E. coli, i.e. EIEC, or EIEC. I would pronounce it EIEC or IEC, or that's how I would pronounce it, but it's entero-invasive E. coli, also called EIEC. Microbe invades intestinal mucosa and causes necrosis and inflammation. So IEC is invasive dysentery. Clinical manifestations are similar to Shigella. What about enterotoxigenic E. coli, i.e. ETEC, ETEC? This produces a heat-labile, heat-stable enterotoxins, no inflammation or invasion. ETEC causes traveler's diarrhea. Enteropathogenic E. coli, EPEC, EPEC. No toxin produced, adheres to apical surface, flattens villi, prevents absorption. Diarrhea usually in children. Think EPEC in pediatrics. That's a good one. EHEC, EHEC. Enterohemorrhagic E. coli. So this is going to include these. O157H7 is most common serotype in the U.S., often transmitted via undercooked meat, raw leafy vegetables. Shiga toxin causes hemolytic uremic syndrome, triad of anemia, thrombocytopenia, and acute kidney injury due to microthrombi forming on damaged endothelium, leading to mechanical hemolysis with schistocytes on peripheral blood smear, platelet consumption leading to thrombocytopenia, and decrease in renal blood flow. You have dysentery, which is a toxin alone causes necrosis and inflammation. It does not ferment sorbitol versus other E. coli. EHEC associated with hemorrhage, hamburgers, hemolytic uremic syndrome. Okay, Klebsiella. Gram-negative rod, intestinal microbiota that causes lobar pneumonia, more common in patients with heavy alcohol use or with impaired host defenses. Very mucoid colonies caused by abundant polysaccharide capsules, dark red current jelly sputum, blood mucus, which is blood, either like blood with mucus, right? It's current jelly sputum. Also cause of healthcare associated UTIs associated with evolution of multi-drug resistance, MDR. The ABCDs, this is a learning device, ABCDs of Klebsiella. Aspiration pneumonia, abscess in lungs and liver, current jelly sputum, diabetes mellitus, and and ethanol overuse. I would say alcohol. But I think your book is a little more reasonable. It says aspiration pneumonia, abscess in lungs and liver, alcoholics, and diabetics and current jelly spew. None of those are, neither is particularly great, but, they have their ups and downs. Just know what it's supposed to cover. What about Campylobacter jejuni? So Campylobacter jejuni is a gram-negative comma or S-shaped with polar flagella, which is shown in the picture. Oxidase positive, grows at the 42 degrees Celsius. Campylobacter likes the hot campfire. Major causes of bloody diarrhea, especially in children. Fecal oral transmission through the person-to-person contact or by ingestion of undercooked, contaminated poultry or meat. Unpasteurized milk. Contact with infected animals like dogs, cats, or pigs is also a risk factor. Common antecedent to Guillain-Barré syndrome and reactive arthritis. Remember, when you hear about a Guillain-Barré, it could be caused by something like Campylobacter jejuni. It could be something else, but this is like a classical example of what can cause Guillain-Barré syndrome. Let's talk about Proteus mirabilis. If it's in here or somewhere. Maybe not. We talk about cholera next. Ooh, that's kind of gross. We'll get to that soon. So let's talk about Proteus mirabilis for a bit. Proteus mirabilis, gram-negative, urease positive, facultative anaerobe, long flagella with swarming motility. Common cause of UTIs, urease virulence factor, hydrolyzes urea to carbon dioxide and ammonia, leading to a net increase in pH. Promotes formation of struvite stones. Significant blockage of renal calyces results in branch stones called staghorn calculi. Very true. And struvite stones. What are struvite stones made out of? Because I got a question on that on a recent test I took on Friday. And I want to know, they say, struvite stones. Well, okay, but what does that mean? Struvite stones. What are struvite stones made of? It should be calcium something, right? Calcium. This is struvite kidney stones are primarily composed of magnesium, ammonium, phosphate. Okay. Triple phosphate stones. Okay. Well, I guess that answers my question, but I'm not quite sure if that really... Yeah, but what is it made out of? They're saying it's mostly magnesium, ammonium, phosphate. Okay, so be it. Vibrio cholera. I just realized something that the book might have some images which are considered maybe a bit suggestive or inappropriate. No, they're not sexually suggestive. They're just showing certain areas of the body that are, normally private, if it makes any sense. So I might have to avoid showing that, perhaps. Well, I think you already saw it on your screen, so I don't know if I can really do anything about it now. But anyways, Vibrio cholera. Vibrio cholera is a gram-negative, flagellated, comma-shaped, oxidase positive, which grows in the alkaline media. Endemic to developing countries, produces profuse rice water diarrhea via enterotoxin that permanently activates G-sub-S, increasing the cyclic AMP. Sensitive to stomach acid labile, requires large inoculum, high infectious dose. Infectious dose 50, right? ID 50. Unless host has a decreased gastric acidity. Transmitted via ingestion of contaminated water or uncooked food like raw shellfish. Treat promptly with oral rehydration solution. Vibrio vonificus, which is a gram-negative bacillus usually found in marine environments, causes severe wound infections or septicemia due to the exposure to contaminated seawater. Presents a cellulitis that can be progressed to necrotizing fasciitis in high-risk patients, especially those with high serum iron, like, for example, cirrhosis or hemochromatosis. Serious wound infection requires surgical debridement. Okay. What about Helicobacter pylori? Helicobacter pylori is a curd, flagellated, motile, gram-negative raw that is triple positives, catalase positive, oxidase positive, and urease positive. It can cause urea breath tests or fecal antigen tests for diagnosis. Urease produces ammonia, creating an alkaline environment which helps H. pylori survive in acidic mucosa. Colonizes mainly antrum of stomach, causing gastritis and peptic ulcers, especially in the duodenal ulcers. Risk factors for peptic ulcer disease, gastric adenocarcinoma, and malt lymphoma, also called maltoma. Most common initial treatment would be a triple therapy, amoxicillin, which is metronidazole if penicillin allergy. So if you use normally amoxicillin, but you can use metronidazole instead if you need to. Plus clarithromycin, which is a macrolide. Plus proton pump inhibitor, so like maybe like a meprazole or something like that. So again, amoxicillin, clarithromycin, and a PPI. Antibiotics cure pylori. Bismuth-based quadruple therapy if concerned about macrolide resistance. Spirochetes. Spirochetes are spiral-shaped bacteria with axial filaments, and they include Leptospira, Treponema, and Borrelia. Only Borrelia can be visualized using aniline dyes, right or Giemsa stain, in light microscopy due to size. Treponema is visualized by dark field microscopy or direct fluorescent antibody DFA microscopy. The little mnemonic or little learning device is little twirling bacteria, LTB, for the three species, Leptospira, Treponema, and Borrelia. So, and we also have this thing called the Jarisch-Herxheimer. I mean, I would just call it Jarisch-Herxheimer, but it's pronounced with it's a J. So maybe Jarisch, but Jarisch-Herxheimer reaction. Flu-like symptoms, fever, chills, headache, myalgia after antibiotics are started due to the host response to sudden release of bacterial antigens. Usually occurs during treatment of spirochetal infections. So this is a reaction you can get as a consequence of treating these spirochetal infections. Jarisch-Herxheimer reaction. It's have this sort of flu-like symptom presentation as a consequence of using these antibiotics, potentially. What about Leptospira my book has that over here, so I'll read my book the way that it's written. Mine goes into Lyme disease first, so I'll go to Lyme disease now. Lyme disease, caused by Borrelia burgdorferi, which is transmitted by the Ixodes deer tick, also a vector for the Anaplasma species, and Protozoa babesia. Natural reservoir is the mouse. Deer are essential for tick life cycle, but do not harbor Borrelia. So the deer are needed for the life cycle, but they don't harbor the actual disease, Borrelia. It's common in northeastern United States. Stage 1 would be an early localized erythema migrans, which is a typical bullseye configuration, as you can see in the picture B. Is pathognomonic, but not always present. Flu-like symptoms can be present, though, for stage 1. Stage 2. Early disseminated secondary lesions, carditis, AV block, facial nerve, Bell's palsy, migratory myalgias, and transient arthritis. And then stage 3, later disseminated encephalopathy, chronic arthritis, peripheral neuropathy. And here's a little learning device here. A key lime pie to the face, facial nerve palsy, typically bilateral, arthritis, cardiac block, and erythema migrans. How do you treat it? Doxycycline, first line, amoxicillin, pregnant patients, children less than 8 years of age, ceftriaxone if IV therapy is required. Very, very, very specific, the bullseye target lesion kind of thing, the bullseye lesion. Very characteristic. What about Leptospira interrogans? So Leptospira interrogans, spirochete with hook-shaped ends found in water contaminated with animal urine. Leptospirosis is the flu-like symptoms, myalgias classically on calves, jaundice, photophobia with conjunctival suffusion, erythema without exudate, prevalent among surfers and in tropics, for example, Hawaii. Wild disease, which is an icterohemorrhagic leptospirosis, severe form of jaundice with anazotemia from liver and kidney dysfunction, fever, hemorrhage, and anemia. Okay, what about syphilis? Oh boy. Okay, let me avoid the pictures for now. You saw them before. They're not great. They're not so wonderful. But if you want to look what syphilis causes to the body, be my guest, I guess. I mean, I would obviously. Well, I mean, you're just looking at different parts of the body that have symptoms of syphilis. It's pretty gross, but it is a gross disease, and that just is what it is. Syphilis. Caused by spirochete, Treponema pallidum, treatment penicillin G. Primary syphilis, localized disease presenting with painless canker. Use fluorescent or dark field microscopy to visualize treponemes in fluid from canker. And you would see that with picture. You know what? I might just show it to you. This is the canker, and this is on a phallus. It's gross. Yeah, I know. Sure. It's a painless canker, though. This is different from Haemophilus ducreyi, because Haemophilus ducreyi, you will have a painful cancroid, but this syphilis has a painless canker. If available, so localized disease presents with a painless canker. Use fluorescent or dark field microscopy to visualize treponema in fluid from canker. VDRL positive in 80% of patients. Secondary syphilis, disseminated disease with constitutional symptoms, maculopapular rash, which is shown in picture B for me, but you see it in picture C for you. You see picture C right there? Anyway, including palms and soles, which is C. My book says C1, C2. You can just see the picture right there for the palm right there. Condylomata lata, which is picture D for me, which is that's on the vagina basically. If you yeah, I'll show. Yeah, that's picture E for you. That's what that looks like. Condylomata lata, which is a smooth, painless, wart-like white lesion on genitals. Lymphadenopathy, patchy hair loss, also confirmable with dark field microscopy. Serologic testing, VDRL/RPR, nonspecific. Confirmed diagnosis with specific test, for example, FTA, ABS. Secondary syphilis is systemic. Latent syphilis is positive serology without symptoms may follow. Tertiary syphilis. Tertiary syphilis is important. Gummas, chronic granulomas, aortitis, vesivosorum destruction, neurosyphilis, tabes dorsalis, general paresis, Argyll-Robertson pupil, which is prostitute's pupil. It constricts with accommodation but does not react to light. Signs, broad-based ataxia, positive Romberg, Charcot joint, stroke without hypertension. What about congenital syphilis? Well, let me show you what a gumma looks like. Gumma is picture F for you, so let's just look at gumma. That's gumma on that guy's face right there, or woman's face. I don't know who it's a man or a woman, but does not look fun. Anyway, congenital syphilis, which is also not fun, presents with facial abnormalities such as ragades, linear scars at the angle of the mouth, black arrow in F, snuffles, okay, nasal discharge and red arrows, F, the saddle nose, no. Let me just show you the picture. Just see. Look at compare. Just look at the description. Picture G. Look at picture G right there. Look at picture H right there. Presents with facial abnormalities such as ragades, linear scars at angle of the mouth, black arrow, which is the black arrow in F for me, but it's different for you. That's G for you. Saddle nose, notched Hutchinson teeth that you can see in picture H. Mulberry molars and short maxilla, saber chins, cranial nerve 8 deafness. To prevent, treat patient with early in pregnancy, as placental transmission typically occurs after the first trimester. It's pretty early on, and that makes it a torch infection because you can get it transplacentally. So now we're talking about diagnosing syphilis. Diagnosing syphilis. VDRL and RPR detects, is this the right category? Yeah, VDRL and RPR detects nonspecific antibody that reacts to beef cardiolipin. Quantitative, inexpensive, and widely available test for syphilis. Sensitive but not specific. Non-treponemal tests like VDRL and RPR and direct testing revert to negative after treatment. Antibodies detected by treponemal tests, FTA, ABS, TPPA, will remain positive. Falsely positive results on VDRL with pregnancy, viral infections like EBV or hepatitis, drugs like chlorpromazine and procanamide, rheumatic fever, which is rare, and lupus like anticardiolipin antibody and leprosy. Now they talk about Jarisch-Herxheimer reaction in your book here. You can read about that there if you want to. It's flu-like syndrome because of the bacteria which is releasing toxins when they're being killed. It creates a flu-like syndrome. They say bacteria, usually due to killed bacteria, usually spirochetes releasing toxins. Okay. Anyway, zoonotic bacteria. Well, my book doesn't go into zoonotic bacteria. It goes into chlamydia. So I'll talk about chlamydia next. Gardnerella, typhus. So many diseases. Yeah, okay, here we go. Is that an eye? I would assume that's the eye. Okay, so chlamydia. Chlamydia cannot make their own ATP. They are obligate intracellular organisms that cause mucosal infections in two forms. The elementary body, which is small and dense, is the infectious and enters the cell via the endocytosis and transforms into the reticulate body. But the reticulate body replicates in cell by fission. Remember, the elementary body is like the early stage, and then the reticulate, which is the one that is infectious, and the reticulate body is the one that actually is mature. So reticulate body replicates in cells by fission, reorganizes into elementary bodies. Chlamydia trachomatis causes neonatal and follicular adult conjunctivitis. As you can see in picture A, that's a conjunctivitis. Non-gonococcal urethritis, PID, and reactive arthritis. Chlamydophila pneumonia and Chlamydophila cystaceae cause atypical pneumonia transmitted via aerosol. Chlamydial cell wall lacks classic peptidoglycan due to the reduced muramic acid, rendering the beta-lactam antibiotics ineffective. Chlamys means cloak, because it's chlamys cloak intracellular. Cystaceae has an avian reservoir parrots. Causes atypical pneumonia. Lab diagnosis, PCR, NAAT, cytoplasmic inclusions, reticulate bodies seen on GEMSA or fluorescent antibody stain smear. Treatment would be doxycycline, azithromycin for pregnant patients, add ceftriaxone if concurrent gonorrhea test is positive. And I would say this, that typically speaking, if a patient has gonorrhea or chlamydia, they will give them both drugs. They will say, okay, we think you probably have this drug, you probably have this disease, and you probably at the same time have the other disease. If you have gonorrhea, there's a very good chance you have chlamydia and vice versa. And so if you have one, they'll give you both drugs. If they'll give you, and let's say you have chlamydia, they will give you doxycycline. They'll give you doxycycline, for example, and they'll say, oh, because you probably also have gonorrhea, we'll give you ceftriaxone on top of that. And vice versa, if you have gonorrhea, they say, okay, we'll give you ceftriaxone, but you probably might likely have chlamydia too, so we'll give you doxycycline. Make sense? Chlamydia trachomatis serotypes. Types A, B, and C. Chronic infection causes blindness due to the follicular conjunctivitis in resource-limited areas. A, B, C, Africa, blindness, chronic infection. Types D through K. We're still talking about chlamydia here. Types D through K, urethritis/PID, ectopic pregnancy, neonatal pneumonia, staccato cough with eosinophilia, neonatal conjunctivitis 1 to 2 weeks after birth. D through K is everything else. Neonatal disease can be acquired during the vaginal birth if pregnant patient is infected. What about types L1, L2, L3? Well, these are going to be lymphogranuloma venereum, small painless ulcers on genitals leading to swollen, painful inguinal lymph nodes that ulcerate bulbos, treat with doxycycline. So the good way to remember is that it's referring, when you have L1 through 3, it's referring to a lymph node issue in the sort of the pelvic region, inguinal lymph node. So when you think about L1, think L lymph node of the inguinal region. Anyway, Gardnerella. I saw that just a minute ago, Gardnerella, or a few minutes ago. Gardnerella. Isn't there so many species you can learn about? So many things you can get infected by. We're not going to mycoses quite yet. Gardnerella vaginalis is a pleomorphic gram-variable rod involved in bacterial vaginosis. Presents as a gray vaginal discharge with fishy smell, non-painful versus vaginitis, which would be painful, associated with sexual activity, but not sexually transmitted though. Bacterial vaginosis is also characterized by overgrowth of certain anaerobic bacteria in the vagina due to the decrease in lactobacilli. Clue cells, vaginal epithelial cells covered with Gardnerella, have stippled appearance along the outer margin, which is shown in the picture in A. Amine whiff test, mixing discharge with 10% potassium hydroxide enhances the fishy odor. Vaginal pH of greater than 4.5 during infection. Treatment would be metronidazole or clindamycin. So now we're going to go into zoonotic bacteria. And now we're on 147, by the way, and we're going to wrap up. Wait a minute. What are we going to wrap up? Okay. So we're 147. We finish our section on 148, and then we'll try to do a short section about mycology, which will wrap up on 151. I think that's probably a fair one to do because it's probably more of the midpoint. I know it's a little bit of a long video, but it's better to have this be at maybe, let's say, 3:20 or 3:30 than have this video be at 3:15 and then have the next video be 4 hours long or something like that. Because next video is gonna be longer than this one, almost certainly. I would imagine. Whatever. Anyway, zoonotic bacteria, zoonosis, infectious disease transmitted between animals and humans. So we have species, disease, and transmission and source. So let's talk about that. Anaplasma species. Species would be anaplasmosis. Disease would be that. So anaplasma species cause anaplasmosis and it's transmitted by Ixodes ticks, which live on deer and mice. Bartonella species cause cat scratch disease and bacillary angiomatosis, and they're spread via cat scratches. Makes sense. Borrelia burgdorferi causes Lyme disease and it's spread by Ixodes ticks, which live on deer and mice. Borrelia recurrentis causes relapsing fever, which is spread via a louse, or like lice, recurrent due to the variable surface antigens. Brucella, which causes brucellosis or /undulant fever, spread by unpasteurized dairy, inhalation, or contact with infected animal tissues or fluids. Campylobacter causes bloody diarrhea. It's spread by feces from infected pets or animals, contaminated meats, foods, or hands. Now, Chlamydophila psittaci causes psittacosis. It's spread by parrots and other birds, and it also causes atypical pneumonia. Coxiella burnetii causes Q fever, and that's going to be spread via aerosols of cattle/sheep or amniotic fluid. Ehrlichia sheffiensis causes ehrlichiosis, which is spread via amblyomma lone star ticks. Francisella tularensis causes tularemia. And it's spread via ticks, rabbits, and deer flies. Now, Leptospira species causes leptospirosis, which is spread via animal urine and water and recreational water use. Now, Mycobacterium leprae causes leprosy, and it's spread in humans with lepromatous leprosy, or in armadillos occasionally. Even though they're the reservoir population, they're pretty rarely give it to people. Usually people get it from other people. Pastorella multicida causes cellulitis and osteomyelitis, and it's spread via animal bite, cats, and dogs. Brixedia prowazeki, is that Polish? Prowazeki causes epidemic typhus, and it's spread to humans via human body louse. Okay. Rickettsia rickettsii, Rocky Mountain spotted fever is what it causes, and it's going to be spread via dermacentor, dog tick. Rickettsia typhi causes endemic typhus and spread via fleas. Salmonella species, except as typhi, causes diarrhea, which may be bloody, vomiting, fever, and abdominal cramps, and it's spread by reptiles, and poultry. Yersinia pestis causes the plague, and it's spread via fleas, rats, and prairie dogs are reservoirs, though. And we're going to talk about Rickettsia species, Rickettsia diseases, and vector-borne illnesses. So off the bat, they say treatment is doxycycline, and your book says caution during pregnancy, alternative is chloramphenicol. They took that little parentheses start out in my book. Okay, it says here rash common. So Rocky Mountain spotted fever. Rickettsia rickettsii species. Vector is a tick. Despite its name, disease occurs primarily in the South Atlantic states, especially North Carolina. Rash typically starts at the wrists and ankles, and then spreads to the trunk, palms, and soles. Classic triad would be headache, fever, rash, which is vasculitis, palms and soles. Rash is seen in Coxsackie virus, A infection, hand, foot, and mouth disease. Rocky Mountain spotted fever, and secondary syphilis. You drive cars using palms and soles. So things that mess with the palms and soles are going to include rickettsia, Rocky Mountain fever, Coxsackie virus A, and secondary syphilis. Okay. What about typhus? Typhus. Endemic fleas, R. typhi. Epidemic, human body louse, R. prowazeki. Rash starts centrally and spreads out, sparing the palms and the soles. So rickettsia on the wrist, typhus on the trunk. Rickettsia on the wrist, typhus on the trunk. Okay, what about rare rash that's rare? Rare rash. Let's talk about it. Ehrlichiosis. Ehrlichia, vectorous tick, monocytes with morulae, which is shown in the picture for B. Mulberry-like inclusions in cytoplasm. Mega is the mnemonic. Monocytes equals ehrlichiosis. Granulocytes equals anaplasmosis. So talking about anaplasmosis, anaplasma vector is the tick. Granulocytes with morulae in cytoplasm. So again, they both have the morulae, but in the case of ehrlichiosis, it's going to be the monocytes, and for the anaplasmosis, it's going to be the granulocytes. What about Q fever? Coxiella burnetii, no arthropod vector. Bacterium inhaled as aerosols from cattle/sheep amniotic fluid. Presents with headache, cough, flu-like symptoms, pneumonia, possibly in combination with hepatitis. Common cause of culture-negative endocarditis. Q fever is caused by quite complicated bug because it has no rash or vector and its causative organism can survive outside of its endospore form. Not in the Rickettsia genus, but closely related to it. Okay. I want to talk about mycoplasma pneumonia at some point. Well, that was quick. Mycoplasma pneumonia. Mycoplasma pneumonia, classic cause of atypical walking pneumonia, insidious onset, headache, non-productive cough, patchy or diffuse interstitial infiltrate, macular rash. Occurs frequently in those less than 30 years of age. Outbreaks in the military recruits, prisons, and colleges. Treatments would be macrolides, doxycycline, or fluoroquinolone. Penicillin is ineffective since mycoplasma has no cell wall. Not seen in gram stain. Pleomorphic bacterial membranes contain sterols for stability, grown on eaten agar. And it shows the little picture there for pleomorphic bacterial membrane. Contains, okay, so pleomorphic you can see in the picture. Chest x-ray appears more severe than patient presentation. High titer of cold agglutinins, IgM, which can agglutinate RBCs. Mycoplasma gets cold without a coat, no cell wall. And so that concludes our clinical bacteriology section. I'll go into mycology, but I'll take a short break. Okay. So where were we? Microbiology, mycology. This is going to be a very short section. We're on 149 right now. It wraps up on 151. So it's only about 3 pages. Microbiology, mycology. Systemic mycoses. All of the following can cause pneumonia and can disseminate. All are caused by dimorphic fungi, cold, 20 degrees Celsius, mold, All are caused by dimorphic fungi. Cold, 20 degrees Celsius equals mold. Heat, 37 degrees Celsius equals yeast. So heat is yeast, cold is mold. Make sense? Only exception is coccidioides, which is a spheroidal, not yeast, in tissue. Systemic mycoses can form granulomas like TB. It cannot be transmitted person to person, unlike TB. The treatment would be fluconazole or itraconazole for local infection and amphotericin B for systemic infection. So if it's really serious, take out the amphotericin B, okay? So let's talk about it. Disease, endemic location, pathologic features, unique signs and symptoms, and the notes. So histoplasmosis. It's endemic to the Mississippi and Ohio River valleys. It has macrophages filled with histoplasma smaller than the RBC. Tuberculent macroconidia on culture. Its unique qualities are going to be palatal tongue ulcers, splenomegaly, pancytopenia, and erythema nodosum. Notes would be histohides within macrophages associated with bird or bat droppings, like in caves, for example. Diagnosis via urine or serum antigen. Blastomycoses. It's endemic to the eastern and central United States Great Lakes region. It is a broad-based budding or blastomyces, same size as the RBC. It will show inflammatory lung disease, or might present with inflammatory lung disease, disseminates to the bone and skin, verrucous, best way I can pronounce it, I don't know how to pronounce it exactly. Verrucous lesions may mimic SCC. It says here, blastobuds broadly. What about coccidiomycoses? Coccidiomycoses, southwestern United States, spherules filled with endosporous coccidioides much larger than the RBC. Disseminates to the bone/skin, erythema nodosum, desert bumps, or multiform. Arthralgias, desert rheumatism, can cause meningitis, associated with dust exposure in endemic areas like archaeologic excavations or earthquakes. Interesting. What about paracoccidiomycoses? This is a little bit of a mouthful. Paracoccidiomycoses, found in Latin America, budding yeast with paracoccidioides with captain's wheel formation much larger than the RBC. Similar to blastomycoses, males greater than females, that's true. And here's a little thing for you. Paracoccidioparacels with captain's wheel all the way to Latin America. You see the little captain's wheel? It's kind of cool looking. You don't want to have it, but, still, interesting look to it. Let's go to the next page. Talking about mycoses still. Cutaneous mycoses. Oh, how fun. I'm going to talk about actually opportunistic fungal infections. So let me go do that. Opportunistic fungal infections. We have that little thing in here? Yeah, we do. Right here. So opportunistic fungal infections. Alba equals white. That's where we get the word Alps from, actually. The same etymological root we have for the Alps, the mountains there, comes the word white. I think Albania is also called that because it means white, because they're in the mountains as well. Albion, which is what we used to refer to Scotland or Britain more broadly, but Scotland more typically, it also has the same root origin point as well. It refers to that. Now, Candida albicans. Alba equals white. Dimorphic, forms pseudohyphae and budding yeast at 20 degrees Celsius. Germ tubes at 37 degrees Celsius. This is a complete detour, but the word, a lot of places in the world that have a word related to the word white in it will have a name because they're mountainous. So like Lebanon is called that because it's Laban means white, and it's called Lebanon because it's mountainous and the mountains are white with snow, presumably, I would imagine. They're called the Alps because they're white with what? Snow because they're mountains. Albania is quote-unquote white with snow. Why? Because there's mountains, right? Et cetera, et cetera. Well, there's only a handful of cases where that's true, but I'm just giving you, those are what comes to mind. Anyway, back to Candida albicans, which is not a country. Systemic or superficial fungal infection causes oral and esophageal thrush in the immunocompromised, like neonates, steroids, diabetes, AIDS, vulvovaginitis, diabetes, use of antibiotics, diaper rash, infective endocarditis, people who inject drugs, disseminated candidiasis, especially in neutropenic patients, as host defense relies on phagocytes, chronic mucocutaneous candidiasis. The treatment would be oral fluconazole / topical azoles for vaginal, nystatin azoles, or rarely echinocandins for oral, fluconazole, echinocandins, or amphotericin B for esophageal or systemic disease. Now, Aspergillus fumigatus. Now, Aspergillus will have acute angle, 45-degree angle, branching of septate hyphae, causes invasive aspergillosis in the immunocompromised patients, especially in those with neutrophil dysfunction, for example, chronic granulomatous disease, because the aspergillus is catalase positive. It can cause aspergillomas in pre-existing lung cavity, especially after the TB infection. Some species of aspergillus produce aflatoxins, including the TP53 mutations causing or leading to hepatocellular carcinoma. The treatment would be voriconazole or echinocandins second line. The allergic bronchopulmonary aspergillosis, ABPA, hypersensitivity response to aspergillus growing in lung mucus associated with the asthma and cystic fibrosis may cause bronchiectasis and eosinophilia. Now, what about Cryptococcus neoformans? Cryptococcus neoformans is a 5 to 10 micrometer size with narrow budding, heavily encapsulated yeast, not dimorphic, positive PAS staining, positive PAS staining, found in soil, pigeon droppings, acquired through inhalation of hematogenous dissemination to meninges, highlighted with an India ink, also clear halo in the F picture, which I don't know if you have that in your book or not. Let me see. Yeah, you do. It's picture F still. And mucocarmine, red inner capsule in picture G, which is also picture G for you. And where was I? Latex agglutination test. This is still for Cryptococcus neoformans. Latex agglutination test detects polysaccharide capsular antigen and is more sensitive and specific. Causes cryptococcosis, which can manifest with meningitis, pneumonia, and or encephalitis, soap bubble lesions in brain, primarily in immunocompromised. The treatment would be amphotericin B plus fluticetazine followed by fluconazole for cryptococcal meningitis. What about mucor and rhizopus species? Well, they are regular, broad, non-septate hyphae branching at wide angles. Wide angles, important to note that. Causes in distinction from the Cryptococcus neoformans, which is a narrow budding, and the Aspergillus fumigatus, which is an acute angle. Remember, Aspergillus acute angle, mucor and rhizopus species are regular, wide angle. So they're a regular hyphae with branching at wide angles, okay? So, sorry, I mentioned Cryptococcus in there. I think it's a mischaracterization. Aspergillus is acute angle. Mucor and rhizopus are going to be wide angle. They cause mucormycosis, which mostly in patients of DKA, diabetic ketoacidosis, and or neutropenia, like leukemia. Inhalation of spores leads to fungi proliferate in blood vessel walls, penetrate cribriform plate, and enter brain. The rhinocerebral frontal lobe abscess, cavernous sinus thrombosis, headache, facial pain, black necrotic eschar in face, may have cranial nerve involvement. The treatment would be surgical debridement, amphotericin B, or isoflugonazole. Pneumocystis jirovecii. Pneumocystis jirovecii, yeast-like fungus that causes pneumocystis pneumonia, PCP, a diffuse intestinal pneumonia. Most infections are asymptomatic. Immunosuppression, like AIDS, predisposes to disease. Diffuse bilateral ground glass opacities on chest imaging with pneumatocoeles. Diagnosed by bronchoalveolar lavage or lung biopsy, disc-shaped yeast can be on methenamine silver stain of the lung tissue or with fluorescent antibody. The treatment/prophylaxis would be Bactrim, trimethoprim, sulfamethoxazole, and pentamidine, dapsone, prophylaxis as a single agent, or treatment in combination with the trimethoprim. Atovaquone. Start prophylaxis with CD4 positive cell count drops to less than 200 in people living with HIV. So here's some pictures. You can see the A, B, and C. Okay, cool. What about Sporothrix shanki? Sporothrix shanki causes sporotrichosis, dimorphic fungus that exists as a cigar-shaped yeast at 37 degrees Celsius in the human body and as a hyphae with spores in soil, conidia. lives on vegetation. When spores are traumatically introduced into the skin, typically by a thorn, rose gardener's disease, causes a local pustule or ulcer with nodules along the draining lymphatics, ascending lymphangitis, as you can see in the picture. Causes local pustule or ulcer. Yeah. My picture is different. So they're talking about ascending lymphangitis, which is what I guess that's what I'm seeing. You're seeing a pustule or ulcer. Anyway, disseminated disease, possible in immunocompromised host. The treatment would be itraconazole or potassium iodide, only for cutaneous lymphocutaneous. Okay. Think of a rose gardener who smokes a cigar and pot. Okay, whatever. But it is rose gardener's disease, and you can treat it with potassium iodide or itraconazole. And that concludes the mycology section of the microbiology chapter. And now we're on page 152, and we'll be going into parasitology, but we will not be covering that today. We'll be covering that in our next live stream video, I would imagine. Anyway, if you stuck around this long, hopefully it was of good use to you. And thank you very much for watching. Have a great rest of your day. Thank you, and goodbye.